This is Drink Talk, where we try to solve the world's problems one drink at a time. Each week in this podcast, the topics of today and tomorrow are discussed while we sample some of the finest libations known to man. Join us. We are your hosts, Brian and Britt.
Check us out. Good evening, gentlemen.
Hello. I don't know if it affects... I see Corey is watching. Corey, if you can just let us know, did that come up as just Britt, or does it come up as Drink Talk is live? If you could let us know. Thank you.
I changed that in the settings, so I think we should be good. I don't
Okay.
know. New technology.
Yeah,
we'll figure it out one
see.
of these days.
Yeah. So we've got some new people. Oh, well, we've got a new person on the program and a return on the program. This is Nick Ted. He goes by both. Yeah, it goes by both. Yes,
Nick
he's
Ted.
a science teacher. We won't say where. We'll just say he's a science. He's going to be the science portion of the
Nick the
program.
Shtick.
There you go. Nick the stick.
So he's going to be bringing all the sciencey stuff because Britt and I are not
We're not
scientists.
scientists, and he is a scientist.
Yeah.
Everybody's a scientist.
And then the back of the head there is Mike, the returning guest host. If you follow the program, you should know him.
Yeah.
Thanks, Corey. Thank you for the update on that.
So, just to start off, we're kind of doing a pre-gamer here. We really didn't get to do a pre-gamer because somebody was running a little late.
We won't say who.
We won't say who.
It was me.
But this comes from one of my good friend Andrew's basement. He is a brewer, and he tried to replicate Pseudo Sue by Toppling Goliath, which we will be drinking on tonight.
Yes, some awesome dude brought it.
Yeah.
Nick Ted brought that. I told him to just bring one of his favorites to the program to share with everybody. But on that note, we've already clinkied, so
Yeah,
I mean,
well, we
so
can still
do it
clink
again.
again.
Yeah.
Why not?
Do
Cheers,
it for the
boys.
cameras, yeah.
Oh, man. He did a wonderful job.
He did.
With
It's-
the hops, and it's juicy. you know?
Very, very close to it.
Yeah.
I would say that you don't get much closer without actually being Pseudo Sue.
Yeah. Kudos to you, Andrew.
Yeah, my music's
Yep.
fine. That's good. So today we'll get into more of the actual beers for the program here in just a few minutes. But while we're waiting for us to finish these, we're going to get into the talk a little bit, and it's gonna. We touched on it briefly on the Gattaca episode, and you were on that episode, I think, Mike, and it was mainly about that legislative bill that was coming in that would allow insurance companies to see your health, your medical history, and all that.
Right.
But we touched on CRISPR in that because CRISPR is used for we'll get into technically, but everybody calls it CRISPR, but it's CRISPR-Cas9 is the whole system that is actually used for gene editing. And to start it off, CRISPR, it's an acronym. It's CRISPR, standing for Clustered Regular Interspaced Short Palindromatic Repeats.
When you first suggested this program as CRISPR, I was like, so you want it darker? You want to put things in the oven a longer time so they become CRISPR?
Yeah,
like
exactly.
I didn't get what you were
We're
trying
going
to
to be
say.
baking on the program. It's going to be interesting.
But when I actually looked it up and checked it out, I was like, mind blown. Because it's interesting what they're doing out there.
Yeah.
Can you do the acronym one more time, like 50% slower?
Sure, absolutely, and I'll kind of break it down because there is some third grade English in there. That so you have clustered regularly interspaced short palindromatic repeats. So we can break that down. So what it is, it's a DNA. When scientists saw it, it was DNA, and in the DNA there were these little repeating palindromatic things. Now, palindrome is a word or phrase that is the same backwards and forwards, like race car.
If you're on Facebook right now, let us know what your favorite palindrome is.
Yeah.
Mine is, was it a rat I saw?
Ooh, that's a wow.
Not too bad. Not
That's
too bad.
that's really well. Yeah, I didn't even come up with phrases. I was just doing words. Man.
Google it. There's some great ones out
Yeah.
there.
Wow.
Yeah.
Oh.
Oh, ding ding.
So Chinese scientists actually saw these, and we're going to get into it here in a little bit. But repeating pairs of DNA, and DNA is the A, C, T, G. They don't repeat at regular intervals. That's rare to do in nature. Usually it's more random. So they didn't know what that meant because this was back in the 80s and we hadn't really sequenced DNA yet. And you can always jump in.
Sure,
you're
the science part.
It so far. Good. Oh, and I guess I could get into the history a little bit. Yeah, so 1980s, and it was Japanese scientists. I take that back, not Chinese scientists. Japanese scientists found this repeating palindromatic pattern in DNA, didn't know what it was, and then fast forward to the 90s when Bill Clinton said that we need to sequence DNA. We ended up spending about $3 billion to sequence DNA, which took 10, 15
It took a long
years.
time for one sequence.
For
of
one
human
sequence,
DNA.
yep. So we have that sequence. We don't know what it all means yet, but it is sequenced. And then in the 2000s, they took a look at it. After we had finally sequenced the DNA, we took a look at that again at the CRISPR, and they said, you know what? That is a virus. So basically, what it did, the DNA took it and took like a photo shot or a mug shot of the virus, and the immune system uses that. Like, hey, we've had this before, we don't want it again, so we're going to incorporate it into the DNA strand, and then once we see this again, then we can send the white blood cells out and attack that. So it's kind of like a little mugshot of virus.
It's like a bit-level digital signature
Right, exactly.
basically.
Yeah, exactly. Which is basically what DNA is anyway.
in
right?
in other nerd speak.
Yeah,
Right.
Yeah. So that in itself was pretty cool, but there's not really a lot we can do with that because the CRISPR by itself is just that repeating strand.
It's like a defense mechanism, basically.
Right,
Yeah.
for your immune system. That's all it is.
It's just identifiable patterns in DNA that happen to be palindromatic and repeat themselves every in a regularly guessable interval.
Right, yeah.
But the thing that like shocked them back when they first discovered it too was that you had these normal sequences, and then all of a sudden you had this just random conglomerate of that
Right.
virus DNA, which back then again we hadn't really sequenced out all
Right.
of that DNA yet, so that was weird to them. They didn't know it was virus DNA quite yet.
Gotcha. Yeah, get up in the microphone there.
Oh, a little closer there.
There you go. If it sounds good in your ears, then it'll sound
Perfect.
Good.
I'll get
Yeah.
intimate with the microphone.
There you go.
I just
Get to
turned
know
you
it.
up a little bit. It's fine.
Perfect.
Yes, but there's not. I mean, other than what it is, there wasn't really much you could do with it. We just
Exactly.
understood what that was.
And it was the scientists had sort of started realizing kind of these larger ramifications, but not to a meaningful extent.
Mhm.
So they went 20, 30 years without really
Right.
finding that significant impact.
Exactly. And then in like 2012-ish, Jennifer Doudna and Emmanuel Compotier, I believe is how you pronounce her last name.
That's close enough.
Yep. So they had this Cas9 system, which is Cas9 stands for CRISPR-associated protein 9, and what it does is it acts as like little scissors in DNA.
Think like Pac-Man is kind of the rough shape, a really pixelated version of Pac-Man.
Yeah, very pixelated version of Pac-Man, and it's very precise. It can cut like single things out of DNA, very very precise. So they knew of CRISPR, and then they said, well, what if we combined CRISPR with the Cas9 system, and it was able to recognize whatever we wanted it to recognize, cut that out, and then once it's cut, then we can add in whatever we want. And so
That's messing with God's plan there.
yeah, the Cas9 system actually unwinds the DNA and then cuts it. And this happened, like I said, 2012, and since then the cost has gone from, I want to say it was like a few $100,000 to now today you can buy it for less than $75.
And almost anyone could do it.
And yeah, if you have a lab at
takes
home,
a-
I mean, it takes a lab,
Right.
But I mean, you can have a home lab and do it.
The-
I wouldn't say a child would be able to do
Right.
it, but
So
Well, yeah, you'd be surprised.
so wait now. Repeat what you just said. I can do what you're describing here at home for $75.
yes.
Yeah.
And then what?
Everything.
And then what do I do with the spliced DNA that I've created?
Well,
Create a dinosaur.
yeah. Yeah.
Your chance.
Jurassic Park.
that's, when was that? Was that 2 years ago? They talked about bringing back the woolly mammoth.
Yeah, they're working on it.
And that's they were going to use the CRISPR system to do so, and they were going to take an elephant and they were going to splice this DNA with using CRISPR, and then add in the other DNA, the missing DNA parts from a woolly mammoth, because we have the DNA of that from hair or bones or whatever that we found, and then they could create a modern day woolly mammoth.
So I guess I'm with
Which Jurassic Park?
What
Did they ever see that movie?
$75 gets me a kit to do what?
It's the CRISPR Cas9 kit.
So you get this little chunk, this little enzyme, and you can program it.
It's a
basically.
protein. Yeah, Cas9 system is a protein.
You just
That
give
is
it,
a
you
programmed.
feed it. Yeah,
It's a programmed protein that you still have to do some more programming in what you want it to find, and then you also have to have the DNA.
Oh, so like if I want to try to find a virus in my system,
yeah,
it'll do that.
yeah,
Good.
I mean, that's
They can do whatever you want.
Yeah, and you're talking right now. They're doing it in plants and animals.
Gotcha.
There are some human testings going on right now, but it's basically very simple to use. And what they're finding is it's pretty much universal.
Right, which is rare for any sort of gene editing tool. The
Yeah.
tools I used just 2 years ago for when we did some gene editing, it was you kind of hoped it worked, and it was expensive.
What?
Um,
Who's we? Who's doing gene editing?
Everyone.
Lots of
What?
because it's so cheap. Lots of people
So my
now.
mind just
And this was
exploded.
before that was before CRISPR became kind of the go-to technology.
Yeah,
And you would still kind of now that everything's sequenced, or a lot of the genes we care about, I
Yeah.
suppose, are sequenced. You just map that gene out, find it, and then you can cut off the ends and then take it, reproduce it, or plug it into something else.
right.
But the technology before CRISPR was a lot more rough. It was really rough. So you kind of hoped it would find the right place, and sometimes, from experience, you would spend 6
that's-
months and several $100,000, and then you would have the complete wrong sequence.
Right, and that still goes on today.
Oh, yeah.
But you're talking. I don't think it's days yet. I think it's weeks that you still have a viable product in weeks instead of 6 to 8 months. You have a couple weeks. The universal part that we were talking about, I guess, should explain a little further. They have yet to find a species across anything that it will not work with. It works in every species that they've done
Anything
that
with DNA.
anything with DNA.
So it's ubiquitous to DNA. If it has DNA, then this technology will
It's going
work.
to work for it.
Yep.
Brilliant.
Yeah.
So I don't want to go down a rabbit hole here.
No, that's what we do on
I
this program.
totally can. But I just want to point out that as the technology nerd
Mhm.
in the house, I hadn't gone real deep on the CRISPR technology and like what exactly it does. I knew that it snipped the DNA.
Well,
and then,
that's what this guy's for.
and then replaced. Like I knew a little bit, right? I knew I looked it up on Wikipedia version, and that was about it.
Mhm.
But
Which gives you a good baseline. But
yeah. But after hearing you guys describe it and talk about it from a computer technology hacker nerd perspective, that's a SQL injection, man. That is a so if any computer nerds that are out there, any hackers that are out there, replacing runtime code is an injection. That's a commonly used mechanism by hackers to take a set of code that's used to execute, right?
Mhm.
Because our body is essentially executing the code written in
yeah.
DNA. And if you can convince the computer in some way to replace a section of the code with what you want in there, then it will execute whatever code you put in there. It's called a remote code execution. You've probably seen it, right? You get a pop-up on your Windows machine that says, this vulnerability could allow remote code execution. That's what we're talking about here.
Don't do it.
Remote code execution is exactly what we're talking about here. We're taking remote code, something that we want, and injecting it into the runtime code
Right.
so that the machine will run the code that we want it to run, not the code that the programmer wanted it to run. The programmer, which we can talk about later,
Yeah.
because that's fun.
Yeah, exactly. So,
That's
so we are hacking evolution.
Yes, you're precisely,
Yep.
yes, you're hacking the human right now.
Yeah,
And with that, we're going to go to our
yeah.
commercial
This
break.
this is actually a good spot to take a break on.
Yeah.
This. So this is our first actual beer. Of
Well,
the podcast.
so I'm glad that we started with Andrew's beer first because I was like, hey, he was trying to mimic Pseudo Sue, and it's funny that you brought Pseudo Sue.
Yeah.
So this is with this is Toppling Goliath. It's Pseudo Sue. Get it now while it's hot because right now they can't make it fast enough. The born on date I'm sure was like a week ago, and you will find it in most stores that way.
It's in a 4-pack.
It's in a
of
4-pack.
tall cans.
Yeah, you can get it everywhere. You can get it
Yeah.
at Hy-Vee.
Love the
You
can.
can get it at Casual
Yeah.
Pint.
Yeah.
You can get it
That's a
at
good
Spirit
spot
World.
there.
You know, I'm gonna. It comes from Deborah, Iowa. So sorry, we're giving our money to our neighbors.
Toppling Goliath is the name of the brewery.
Yep. So it's just about 2-3 hours up into Iowa. I would love to take a field trip out there. They've got awesome artwork on their cans. There's a giant dinosaur on it, a giant T-Rex on there.
Which is another good tie-in to what we're talking about
Exactly
Ooh.
why I picked it, because
here. Right?
soon it will be off the can.
I know,
no
we're-
diggity,
Walking
no
down the
doubt.
That's like a Jurassic Park tie-in.
Oh, yeah.
Which You know.
So with that,
my
cheers.
5-year-old would love this beer.
Yeah.
Can. The can.
And I
Beer
believe
can.
that it's a 6.5 or something like that.
I actually didn't, we've had it on the program
Yeah.
before, but I actually, because I didn't know until I got here, I didn't do any research on it again for the program.
So this, compared to his other one, it's just a little more crisper.
Weird.
Right?
Yeah.
It's a little more crisper of a flavor. It kind of hits the back of your throat a little more.
But he's got the foundation right there.
He's got the fountain.
Yeah.
He's got the
Crazy.
juiciness. Oh yeah.
And I guess we should talk about the flavor profile. I mean, this is it's a citrus forward IPA is what this is. And so those of you that are fans of citrus or IPA, this is a really good one to it's one of my go-tos. And actually Britt bought it for me yesterday. I had a
Yeah.
Had a
What
can
did
of
I got you this one, the Pseudo Sue, and then I got you the other one. I can't remember the other.
Pompeii.
Pompeii. That's
Yeah.
right.
Mhm.
Yeah. Gave him a little holiday birthday 4-pack thing.
Yeah.
Can't
yeah.
go wrong.
What he doesn't know is I took the other two from the other pack and made myself a 4-pack.
Nice. Oh, whatever. I guess we can. So we should talk about what's being done. Unless you have questions about CRISPR, did we explain that fully?
Yeah, it's.
I mean, you know
you're,
it.
you're
Basically,
taking
yeah.
DNA and you're chopping DNA out and replacing it with better DNA, or taking it out altogether.
And from what I've heard, it's actually relatively easy. Like once you snip that little portion of DNA that you want out
Yeah.
of there, like you just have to get the other DNA just
Yeah.
like close, and then it's like, oh, hey, and grabs it and like sucks it
Well,
in.
it's I don't want my son or daughter to have.
Snipping their own DNA.
Well, I don't want them to have blonde hair. I want them to have brown hair.
Oh,
see, you're getting into the way.
Well,
yeah,
I mean, that's
give me. Let's talk. Yeah, let's talk about what we're getting.
This is
what
not
we're doing.
spoiler alert. I mean, it's what we're doing.
This is
We're
a
cutting genes.
gradual build up here.
One more quick point on the hacker thing that I said. The RI in the CRISPR, the regularly intervaled, is really key to the success, I think, from a biology perspective, at least in any technology and any computer hacking that I've ever done. Having something that is predictable allows it to be hackable. If I know what that computer is going to do or what that code is going to be, then I can trick it. Then I can trick the machine. But it's that predictability that allows it to work. You have
Right.
to be able to say, I know what this thing's about to do, so you can preempt it. Like with the Cas9 thing, you have to be able to tell it, look for this, because we
Correct.
know it's going to do this, and when that happens, then do your magic.
Yeah, and
Right, it's that
if you don't know how to do that, it's not like a DOS prompt. It's like
Right, yeah, right.
You got to actually know how to hack into this Cas9 system.
Well, and that's
Sure.
the big push, or I guess.
I wonder
the
if there's a
big-
YouTube video.
Oh, I guarantee you there is.
Yeah.
But that's the big reason why the sequencing was so important. So we had all this. We could roughly tell what's going on. We could roughly tell that CRISPR in the 80s was this defense mechanism for bacteria. But outside of that, we didn't really know what or why because we didn't have that genetic information. And like you said, we spent, $3 billion on the
I
in
think
vivo
of that.
project?
Yeah,
it was
roughly.
high.
Yeah.
And we can now, $70 for
Yeah,
ancestry DNA. Everyone can do that now. So
Right, well, and that's another point. Kind of what you were talking about with the repeating patterns. Another point to this is the cost of it, because of the way technology moves. It gets cheaper as time goes on, but it is so cheap right now, and it's relatively new that everybody's doing this. Like scientists are going crazy over this CRISPR-Cas9 system. And it's so inexpensive that anybody can do it, which innovation and patents are just exploding right now because so many people are doing it.
right.
So,
And who allowed this? Who was the president? Was it Clinton? that allowed this?
so Clinton sequenced the DNA. This isn't something that
I'm just wondering. When did this all get started? What kind of year?
2012 was when that duo Jennifer Doudna and Emmanuel Compartier.
Okay,
Sh-
2012.
yeah, something like that.
All right, so
And that's when they kind
under
of-
the
yeah.
Obama,
Yeah.
which is
But it
I
has
just wanna
nothing.
know
Yeah, it
what
has
was going
nothing to do with politics.
Well, I
it's
just want to know
science.
who approved this
Well, so
to
it-
be okay,
and-
because
right.
somebody has to say that
Oh, yeah.
it's okay to do this.
Well,
Sort of.
depending on what. Not
Sort of.
Yeah, because we're not doing it on humans yet. I mean, we're doing it in human trials now. But we aren't actually creating designer babies.
Let me
We're gonna
calm
get
the
there.
nerves here
Yeah.
from a science perspective.
Yeah.
So what happens is you have a science ethics board,
Mhm.
and they're hyper strict. So these are people that their entire career is based on understanding and working on the ethics of science problems. It would be completely, gosh, I don't want to, what's the right word? Not useless. Inundating to have to go to Congress each time we discovered something to say,
Right.
can we research this?
Right.
So we have these ethics committees, at least in the United States and a lot of other developed countries, and their primary job is to say, yes, continue doing this, or no, we're not ready for that yet.
And if
or,
it's big enough, then they bring it up to Congress.
right, and so I don't want to jump the gun on the ethics part, but
Yeah.
I know you watched that Netflix video.
Yeah, and that's so. I guess I should backtrack a little bit on how I came about this. I mean, I did touch on it, on the Gattaca episode, but for those of you that are wanting a quick video, and those of you that are Netflix, look up the series Explained on Netflix, and there is one episode. What is it? It's like 20 minutes.
They're
if that.
super quick.
Yeah, and it's called Designer DNA. You can look it up, watch that really quick, have a much better understanding, or you can do a YouTube search on CRISPR, and there's a lot of videos out there about it, and they do a very good job. Hopefully, we do a good job of explaining this,
That's right.
because I've watched a lot of these, and I'm trying to disseminate it and bring you the Cliff Notes version of
Right.
everything here.
It's a lot to take in.
It is.
When you sent me a few things to do some research on, which was thank you. I had a lot going on, so that's great that you sent that to me. When I was watching it, I'm just like, I should be taking notes right now.
It's kind of it's daunting to see because anyone can kind
Well,
of piece
just-
together the puzzle.
To take that little bit of strand of DNA that's so microscopic and go, oh, there it is,
Oh, yeah.
and just cut it right out.
Mhm.
You
Well,
know?
then the astonishing
Well,
thing
if you
is to
think
think
it-
about
this-
we've been doing it for 50 years.
Oh yeah,
Like, oh my gosh, people were doing this before YouTube, before the internet,
right.
before computers.
Hmm. So I guess we should talk about what we're doing now. And actually, a lot of the information that I got from this portion of the podcast was from another podcast called Radio Lab, which is a pretty cool podcast,
Phenomenal.
actually. And they actually did this back in February of 2017. So some of the information is a little dated, a year. And because, like I said, everybody's doing this, it's changing so fast right now. But they're using it in pretty much everything. Is basically what it is. The nuts and bolts of it. Everything it's being used in everything. Crops, medicine, research, livestock, and now humans. They're doing trials on, I think, macular degeneration. They didn't say. They said it was a disease that caused blindness. So I'm assuming.
Are you saying that someday that humans are going to be GMOs?
Yes.
Oh yeah.
Yeah.
Oh,
Yeah.
crazy.
And really, I mean, we are an organism that can be genetically modified. Yes.
Sure.
Well, we
GMO.
pump GMOs into our body once a year.
Yeah,
Flu
we do.
vaccine.
Yeah.
Exactly.
theoretically.
Yeah. So we are already.
Or if you eat at McDonald's,
Or you have
one
to
of the
eat
two.
at McDonald's.
Whatever.
I'm well preserved.
Yes.
This like the eye trials right now. They basically they're talking about injecting CRISPR, taking a syringe of CRISPR and just injecting it into your eye, and then it
Fix your eyesight?
fixed the macular degeneration, which causes blindness.
Wow, that would be awesome.
Yeah. Another one is they actually cured muscular dystrophy in mice, which muscular dystrophy is, and correct me if I'm wrong, it's basically when your muscle, it doesn't turn to fat, but it's like a fat-like substance.
Right. It really degrades.
Yeah, and you've got no strength in it and all that. And what they found in these mice was once they injected CRISPR in there and said, and basically what muscular dystrophy, you lack one protein that basically just turns your muscles to mush, and it replaces the CRISPR system is replacing this one protein. And what they found was that relatively quickly their muscles started getting stronger and stronger.
So help me out here. So for every one of those that you're describing, for every instance of a CRISPR-Cas9 miracle, there has to be a specific, you have to like you mentioned, you have to program the Cas9 to identify that specific segment that you want to replace. And so we have to know what that is, and that's where the human genome sequencing comes into play.
Correct.
So we have to know the
What
human
it does.
genome to know what we want to replace,
Yep.
and then program the Cas9 to work with the CRISPR system to find that specific thing that we've identified, and then go replace that specific thing. But if you're wrong, then what happens?
And
it does happen. Like that does happen still.
And that's why they have they did pass some legislation. Pretty soon. I would guess.
I thought it was a
2012.
self-imposed is it moratorium? Is that the right word? The pause
Moratorium.
Yeah, the
Moratorium
self-imposed moratorium
on several
Hum-
aspects
on human embryo.
specifically.
Yeah.
So of course, I think 4 or 5 months ago, China's already tested on human embryos.
Yes, and I do want to get into that, but I want to wait until
Sure.
the end. So yes, they have, but I want to talk about that a little bit later. There's a couple more things I wanted to bring up that we're currently doing. In 2016, so just 2 years ago, human trials started for certain types of cancer. They want to get rid of lung cancer, and I can't remember the other one. There's 2 that they're specifically working on right now. And basically, and they're doing it in like ingenious ways. What the CRISPR system
Well,
is.
if they're going to get rid of lung cancer, I mean, might as well just get rid of cancer. I mean,
so
do that.
you know what cancer is, Britt? Cells behaving abnormally.
Sure.
That's
Mass
what cancer.
reproduction.
Yeah. So I mean,
But they should be able to detect that, in modern science now.
yeah,
so.
but that's a very generic term.
Well, so that's what I was just going to say. Is so
So
you
here-
threw me when you said cancer, because then you have to imply or presume that cancer is like 100% hereditary.
Right. Okay, so what they're
Is
doing
that accurate?
what they're doing. The CRISPR isn't attacking directly the cancer. It's not going after the cancer directly.
Right. It's replacing DNA.
Nope. What it's doing is it's going after the white blood cells, and it's taking the leash off the white blood cells so they can vigorously attack the cancer way better. So basically, they're giving steroids to our white blood cells.
I see. You're not targeting the disease. You're targeting the human immune system, which is a pre-programmed thing within our DNA. That
Correct.
is pretty
Pumping
well
it
known.
with steroids. You're just
Basically, yeah.
That's
thing in overdrive.
crazy.
Yeah.
The challenge with cancer, especially, is they're basically normal cells, basically, except they're cancer. You have they're mass producing extremely quickly, and everything else looks like a normal cell. So your body doesn't necessarily see this as a threat. It just thinks, oh, the cell's producing, but everything looks normal in the cell, so I'll just go on my business. And then, cancer does its thing. So having a system like CRISPR where you can specifically target or allow something like a white blood cell to specifically target these cells is a huge step in the right direction, at least for solving cancer.
Like I said, I like the way they're approaching it instead of going after the cancer directly, because all cancer looks different.
Yep.
So whenever you talk to a cancer patient, one of the things that you generally talk about is, hey, it's their white blood cell count. And this would be a way to kind of manipulate and artificially, sort of artificially, I don't know,
It is.
boost that white blood cell count.
Yeah.
Or at least
that
make
would-
the
yeah,
ones
that
that
would
are
be
there
good.
stronger.
Positive.
Beautiful.
Exactly.
Check it on the pros. We need like a little
Another-
tally sheet.
yeah.
Pros.
Another one that's interesting, especially now because everybody's on the antibiotic, the superbug thing. What they want to do is they want to replace CRISPR, want to replace antibiotics with CRISPR. So you would take a pill with CRISPR in it, and it would it would be programmed for whatever you have, and it would seek that out, and it would just snip DNA but not replace anything, and so it would just die. So the virus would just
When
go
we
away.
take most antibodies now, or antibiotics now, it's going to wipe out most of your
even things you
bacteria.
need. Yeah.
But we have extremely important gut biomes with tons and tons
Flora.
of bacteria that help us.
Yeah.
Yep. And so that we become sick. It's harder for us to digest. We have
Mhm.
diarrhea and all the fun after effects
Exactly.
of
These,
being sick.
yeah, the after effects are, what is it? The like drug commercials are like half of what the drug
this
is and
list,
half
yes.
of all the side effects. Yeah.
Oh man! But yeah, with CRISPR, all of a sudden you're targeting
This one
specific
comes
ones.
with a puppy.
yeah.
Oh, hey.
Hey, all right. Another one that which is kind of cool, but starts to get into some wow, should we be doing this? They want to cure malaria. So what they want to do,
Well, why wouldn't you?
yeah. Now, if you inject CRISPR into a mosquito to cure malaria, then that mosquito, and it cures it in that mosquito. Well, what they're trying to do is not allow it to,
So you
allow
have Joe
malaria.
the mosquito.
Yeah, you have
floating
Joe.
around.
Yeah, you have Joe the mosquito, and you don't want malaria to be in that mosquito. So you make make an environment within the mosquito that can't thrive or can't stay in. Then it reproduces with another mosquito that doesn't have CRISPR in it, and then now its offspring has a 50% chance of having CRISPR. So what they're talking about doing is what's called gene drive. And what it is they want to put CRISPR on autopilot.
Gene drive is an interesting way to say genocide.
Yeah, it sounds like what they
nice
did in
way,
Brazil.
right? Yeah.
You know,
It actually, when I was listening to this or reading about it, it actually reminded me of your story about nanotechnology. You know, nanotechnology, and I'm going to pose a question here in a minute that is going to be interesting on nanotechnology, but all it's going to take is one wrong sequence of code in nanotechnology to end the human race. Now with this, the potential is very real. What they're talking about doing is injecting CRISPR into a mosquito that basically has autopilot, and it knows what to do. So when it mates, basically instead of having 50% go to the offspring, you have 100%. Like it's already
it is going to go.
It's on autopilot already, so it's going to seek out and it's going to make that offspring.
So those aren't offspring; those are clones.
No, not
I mean,
well,
that's kind of what
it is.
offspring
It is
are.
offspring. It's just that one. It's just the CRISPR systems in that one also at 100%.
Let me know when that's going to happen. I'm going to stay away from Africa for a while.
it's not a 100% copy of the DNA. It's 100%
Not
of
saying
the
we're
CRISPR
doing testing there, but
goes with it. I see.
Exactly. Yeah.
Well, they will be.
Yeah. So it's CRISPR goes into the second generation of mosquitoes at 100%, and it's already on autopilot, so it knows what to do. And then when that second generation makes the third generation, it still has 100% in there because the CRISPR is still at autopilot and knows what to do, seek and destroy.
Yeah, but that's a real. I mean, I joke about it, calling it genocide, but it is, right. You're changing the DNA, the foundational components of a species, and making it something else.
Well, I was just thinking,
Getting rid of that species entirely.
if that happens, I'm
Well, they're not
moving
talking about
underground.
getting. I mean, that would be awesome if they got rid of mosquitoes, but
they're going to get rid of that species. It'll be a very, very minute change in that species, but functionally that species is gone and it has been replaced with a new species.
What I want to know is if we finally get it to where malaria does not thrive in mosquitoes, then where is it going to thrive? Does it go somewhere else?
Does it mutate?
Does
evolve into
malaria
another
mutate?
form?
Does it move into something else? And now we have to worry about the swine flu. You know,
We're
now,
basically taking natural selection and then just putting it in overdrive. What took millions of years in the past, we can do within a day or two.
right.
Well, and like I said, if this ever happens with us mosquito thing, I'm moving underground. That's just what I'm going to do.
Calm the nerves one more
If they
time
get rid of mosquitoes, I'd be happy. But
So what they can do,
Yeah,
or what they're also
what are
doing,
mosquitoes good for?
yeah.
food for other animals.
Unfortunately,
That's it.
I read that the other week.
But
Yeah.
there's other
Everyone's
food
so
out there. in the
desperate to
sea.
find it. Yeah, can you just eat a fly, please?
Exactly.
And move on with your life.
Right?
It's a lot bigger, more nutrients.
Exactly. But to the kind of the counterpoint of kind of forcing that gene into the offspring, they've also found a way to put a timer on these genes. So part of the big problem, which you've mentioned multiple times, is what happens if something goes wrong. Let's say this exact gene we replaced stops malaria from forming, but all of a sudden opens this gate
Pandora's
to some
box.
giant virus that's going to wipe out the entire species.
Now it's in bears.
Exactly. Right. Great. And now bears are rabid and going to eat all humans and proliferate faster.
Yeah.
You never know. And that's the big thing with genetics is
It
there's
attracts
so
bears.
many.
See what you put the office in danger now.
yeah.
So I just want to point out, like, again, I'm the computer nerd in the room, and I've worked with software developers like my entire career. I'm married to one, right? We're talking about manipulating code here. That's what we're talking about, right? And making changes in a production system. Everyone who's a software developer just laughed, right? Because the one thing you never do is make changes to a production system. Do that in your test environment. Okay?
Right,
Yeah.
right, right.
We don't have a test environment, right?
We are the test environment.
Yeah, we're the test environment. So you're making changes in prod, which you don't want to do.
in real time.
And no matter how confident you are in your code, I will tell you this: every single month, every single software developer at Microsoft is largely confident in their code. And every single Tuesday, the first Tuesday of the month, Microsoft releases massive kit of patches to fix their code for the last 20 years because humans aren't good at writing code. And so for us to sit here and manipulate it is, and I don't care
We're
how confident you
infallible,
are
yeah.
in your timer and everything, like,
someone's
wrong.
going to screw up somewhere
it's
down
gonna
the
go
line.
wrong. Somebody will fat finger that
and
thing.
then take
man.
that times a billion, because we're not just talking about one computer here. We're talking about millions of other different types of computers that are all interacting with one another in our ecosystem. So it may not be prevalent to us, but it could wipe out a whole other species. I mean, you just can't.
Yeah.
There's no way to predict that. Yeah,
you
no
can
way.
do your best. You can do trials as much as you want, but there's always that chance that something could go wrong.
Mm-hmm.
Yeah,
Absolutely.
Microsoft could write a bug that takes down the whole internet, and someone could fat finger their CRISPR-Cas9 code and take down the human race.
Yep.
Right. And that points
whoops.
to that
escalated
Sorry,
quickly.
guys.
I apologize.
no that pointed to that story that I was talking about with the nanotechnology. So the question that I bring to you is, with this CRISPR system, because it's already available, it is super cheap and super precise. Are we going to need in humans, I should say, are we going to need nanotechnology? Because nanotechnology can be used for other things.
Oh yeah.
But I mean, within organisms, are we going to need nanotechnology?
Is there a nanotechnology? Are you on the nanotechnology side?
Oh, I mean, I'm a nerd, so only to a small degree. But
Yeah.
But
What was that movie with nanotechnology in it? Like,
With Johnny Depp,
yeah,
and
it
it was
totally enveloped his whole life. He became
Transcendence.
There you
Oh,
go,
yes.
Transcendence.
Yep.
Wow, that was deep.
I would say, will we need nanotechnology? Absolutely, for completely other reasons,
Right.
other than what we're talking about here. But related to this, I would still say yes, because when it comes to a delivery mechanism for CRISPR-Cas9,
Mhm.
if you want to get your CRISPR-Cas9 to a specific area of
You're
the
gonna
body,
need
or a
that.
specific nanotechnology would be probably
Yeah,
an ideal delivery mechanism
Or a
for that.
syringe.
Yeah, see, that's the catch.
what
It
are we
takes
really
one
doing
next,
syringe and
Brian?
one embryo. And
Yeah.
the cool thing, I guess, depending on where you're standing,
Oh, fairy nectar, Britt. Sorry.
thing about Cas9, though, and the CRISPR gene, is that it's going to find that chunk of DNA
No matter
no
what.
matter what. But again, now you're messing with embryos,
Yep.
which again is why
Now you're messing with God's plan.
exactly.
All right, and that's gonna get so. You're. How are you doing?
We're
Getting
doing all
there?
right.
Because I'm at the break point. There's
kinda-
between topics right now.
had a long day.
yeah.
We'll hurry.
Take it
Yep.
down.
One
a
more
notch, Brett.
point, though,
Okay.
if I could.
No, absolutely.
Sure.
Yeah, go ahead.
So we can kind of force this gene to jump into the progeny, right? So it's going to go into the next of kin, and it's going to go into the next and the next. We can make it infinitely progress
And not degrade through the generations.
and not degrade. And counter to that, we can do the opposite. We can say, after 5 generations of this mosquito, it's going to stop no matter what.
Mhm.
So if something happens, this catastrophic event, we know that in 5 generations, it's out.
Unless
We're
it
going
just
ideally
unless
back
it
to
murders
normal.
all mosquitoes.
Well,
Unless the mosquitoes are that good in 5
We've been talking
generations.
about the mosquitoes. Why can't we talk about the bees? Come on, can we
Or
do
the
that to the
bees.
bees?
Well,
you
that's
know,
true.
we need the bees.
Yeah.
But think about it, CRISPR, one of the big problems with the colony collapse for bees
Yep.
is
up in the mic.
the pesticides. Yeah, I'll just eat the mic.
It's all right. It's fine.
Is pesticides, and one way you can avoid using pesticides is GMOs. The other way is, and I mean, this just kind of
that's what they're
encourages
doing.
That, yeah,
I mean, CRISPR is being used in crops to
to prevent.
Yeah.
Yep.
Absolutely. There are pest-resistant strains of certain crops that don't require pesticides.
Right. And you
Already. That's like now. You can
Right.
purchase those now.
And thankfully, that's a lot easier to test as far as health concerns go, because you can feed it to a colony of mice and see if they die or not. And mice have a really similar genetic makeup to humans,
Yeah.
which is why they're the go-to genetic.
You know how
model.
long that's been?
That's
it was-
embarrassing.
We've been
Right.
using
We're so close to
compliant.
mice.
Yeah.
we've been using
almost there,
mice
guys.
since 1974. That's when they became the
Really?
staple.
74?
1974.
I would have thought it would have been like 1930.
as the staple.
Oh,
so
gotcha,
like
okay.
as the
Yeast
staple.
are still up there.
I'm like, I
surprisingly.
thought it would have
Yeah.
been way sooner than that.
Well, but yeast are more basic.
Yep.
Whereas that's a prokaryote, and we're eukaryotes. Is that right?
Oh gosh,
Boom.
put me on the spot.
Science teacher.
I failed.
Oh man. So yeah, prokaryotes are more like the single cell organisms, bacteria, and all that, and eukaryotes are multi-cell.
Right.
Is that correct?
Yes.
Okay.
Not yeast. Sorry. flies.
Okay.
Fruit flies are actually even
Okay.
a better model sometimes for humans.
They multiply faster.
because they multiply faster, and
Yeah.
they again still have a very close genetic code to us.
Yeah.
So
And if you use fruit flies, you can have a 24-hour lifespan, and
exactly,
there you go.
see if they wipe out
Yeah.
human race
Exactly.
a little faster.
All right. So, are we moving on to the next one? Is that the final point that you wanted to make on
I think
on
so.
all
I think
those
that's
questions, concerns, comments before we get into the next
I'm
portion of
super
the
excited
program?
to talk about the ethics.
Yeah, that's where
Like,
we're heading.
Man,
Ready to do
there's
that?
so much.
I know, right? And it's going to be fun.
Yeah, I got you.
Ethics is fun. So, this next one,
I'm a very good pourer.
Me
Yeah.
either.
No, I am.
Oh, you are.
Yeah.
I am not. So this is up
yeah.
to
So this next beer, this one is the
It's
one
all
that I
about
brought.
tipping the glass. So the proper way to pour is if you tilt your glass and tilt with the beer.
you. I will always find a way to mess it up.
It's
Ooh, I got one more for you.
So an actual, well, and with so there's a little bit of head in
Yep.
here, and that's something we talk about. That's
you do
fla-
want a little bit.
Yeah,
Yep.
that's aroma and flavor right there. So the ones that remember in like college you'd pour, not that anybody drinks in college, right? Yeah. So when they pour with no head, because that's what we wanted. You didn't want
Right.
any head.
Should've
If you
said that.
if you
For a
had,
good beer.
yeah, we're talking beer, Britt.
Yes.
So
That's why I was kind of stopped where I was going.
so yeah, if you poured a little bit of head, people would make fun of you. You know, like,
Right.
oh, you don't know how to pour a beer. That's where the flavor and aroma it like helps the beer open up. It's like wine, man.
It's like it's gauging the amount of that. It's kind of like CRISPR. You just kind of hope it works, and then all of a sudden you have.
We
half
put
a
it in
beer full.
autopilot and
Exactly.
Yeah.
go on
And
your
now
merry way.
everyone's beer is going to be
Exactly.
half. Well,
We'll do this.
So,
almost
Although, if we're going down that road, let's talk about the likelihood of using CRISPR-Cas9 to modify like hops and yeast and
I'm sure it's already being done.
and all the brewer's ingredients
That's
to make
what
like
I
the,
did for
the
my,
world's greatest beer ever.
undergraduate
so,
research.
and that's gonna so.
Seriously?
Oh yeah.
Well, while this is settling down, let's talk about that for a second. So, if
He
you're
said
talking,
he did it for his undergraduate research.
and I missed that. I'm sorry.
Oh, you're good. No, yeah. That's
So
I'm
that
so into it.
That poses a whole nother question. So like right now, as a, I'm a whiskey beer guy. Those are my two favorite. So for me, like hops, I love hops. And right now, Washington State and Oregon are the two top producers of hops just because of the climate. CRISPR in hops would completely change that demographic. You could grow it wherever.
right.
And in states like here in Nebraska, we've got a lot of land. So, but we don't have the climate that is preferable to hops.
Right.
We can grow hops here,
Just
but
not as efficiently.
exactly.
But with CRISPR, you could grow it in the tundra. You could grow it
Oh,
in
yeah.
the middle of nowhere.
Yep. Yeah.
You could grow it,
Is that true? Really?
What?
You cut that part out of it that says I'm going to die during wintertime. And
What part of the
boom.
plant do you cut out that says it needs like water and sunlight, right, and nutrients from the
No,
ground?
no.
If you
It's
sequence the DNA, you
during
know.
winter, though, during winter, there's sun and there's light.
You can find
You know,
ways to
there's water.
Square watermelons, Mike. They have square watermelons.
anything can be done. It's the
And
future.
purple. Have you ever seen the purple ones? Those are
No.
awesome.
Well, yeah, and what was it? The first modified tomato was the Flavor Saver back in '94-ish, '94 to '96, somewhere in there.
Don't eat them.
Well, yeah, homemade.
Yeah.
Or homegrown is the best.
I've got
All
four
right.
different tomato plants out there.
All
Get
right.
the real stuff.
On to this next one here. We are so in this talk we're talking about DNA strands and strains.
Yes.
So I thought Kros-Strain Brewing Company would be
Oh, now it makes sense.
Mhm.
I'm like, how is he tying this in?
yeah, and also because this is kind of like black magic, I went with the fairy nectar.
So
We could create fairies with CRISPR.
that's what I was just going to ask. Like, legitimately,
Okay,
can you do that? Like, could you? If we got
Take
to
the wings of a bird and put them on a human.
Well, I'm
no,
just saying, like-
no, and here's why. And
it's
I
a
know,
Pegasus.
I know I know this answer.
Who doesn't
Well,
want
so my
Pegasus?
question is
Right.
like, how far does this go? And we're going into the ethics thing a little bit
Yeah.
here, but like, how far down the rabbit hole can you go? I'm thinking of like Moore's law, right? Because this is going to increase exponentially our understanding of it and our ability to do it and the cost effectiveness of it. And so, where does it? What are the limits of it?
Mhm.
How far can it go? And can we make a fairy?
Yeah, okay, I want to answer that, but let's talk about the beer, and then I will directly answer that because I actually have
Yes.
All right.
one that directly
Jam on
answers
it.
All right, so this is Fairy Nectar by Kros-Strain. This is not the London hop, double hopped, or the triple hop. This is just the
London hop is so good.
It is. This is just the regular Fairy Nectar by Kros-Strain Brewing Company,
Which is local, right here in Omaha, Nebraska.
local. Yeah, well, it's in La Vista, but which is
Whatever.
a
It's
suburb.
Omaha.
It counts.
Yeah, it does. 6.2 ABV. So
The
in
money stays in
mid-range.
Nebraska. It's local.
We're gonna annex. don't worry.
But it is one of my favorite beers. I dig this one. There isn't much that Kros-Strain produces that I haven't liked. Everything is amazing that
I
they
haven't
produce.
had a bad
So,
brew from them yet.
On that, we're gonna clinky.
Yeah, cheers. Cheers.
Cheers to the fairy nectar.
In the future,
it's an
dinosaurs and
And fairies.
Oh.
And it's an IPA, by
Dinosaurs and fairies and hobbits and
you know
Hobbits.
whatever else you want.
It's like
the way. Oh, this is.
it like sizzles as it goes down your gullet. It's like
It's not as so. The last one we had, the Pseudo Sue, was more of a citrus forward.
whoop,
This one is not a citrus forward IPA. This is just kind of the.
hop in your mouth.
hmm
Oh, that's kind of good. Hop
So
in your
they
mouth.
probably don't use Cascade or Citra hops because the Citra hops are
brew it, Brit.
yeah.
Yeah, right.
Mm,
Hop in your mouth.
Brit. Oh. Now I'm going to start a brew company. Brew
trademarked.
Brit.
Yeah.
Trademark.
All right.
Exactly. Happened right now.
Everybody like theirs?
It's
It's
very
really
good.
good.
It's
All right.
so
Have
tasty.
Have you had Fairy Nectar before?
No, this is
No.
new.
It
All
is
right,
so
sorry.
good to drink.
I didn't comment because I'm very familiar with the fairy nectar.
Yeah.
Oh, yeah.
We've met.
Didn't want to assume for everybody. Okay, so to directly answer your question.
Okay, ready to
Oh,
go.
and where did you purchase said alcohol from?
Oh, and I totally forgot that story. Okay, so this morning, or so after church, I went down to Spirit World today. And because they were having
you called me from nice.
So they were having tasting the Lonely Oaks Distillery from Erling, Iowa, which is near Harlan.
I love
Iowa.
those guys down at Spirit
Yeah.
World. They've been on our show a couple times.
Alzeri was there. I talked with him.
Nice.
And I also got to meet the Pat and Amy from Lonely Oaks Distillery. Great people. They've reached out to us.
Sounds like a radio show. Pat and Amy from Distillery.
Who doesn't tasting right after church?
Exactly.
I know, it's
Well,
great.
so what's
Natural
cool about them is they are
Praise the Lord. Going to go get some alcohol.
they're a grain to bottle distillery. So they're farmers.
that's
They
great.
farm the land. They actually plant the crops, farm the crops,
That's great.
create the mash, distill the mash.
They
and
want to
then-
get us out there, don't they?
They do. They
Yeah,
want us to do
nice.
the podcast from there, and great bunch of people. It's a husband and wife that own it, and good group of
Scott,
people.
Scott and Amy?
No, Pat and Amy.
Pat and Amy.
Yeah, that's
Pat
right.
and Amy in the morning.
So they actually had a peach vodka that they mixed with lemonade. Phenomenal.
What did it say that again?
A peach lemonade or peach vodka that they mixed with lemonade.
Oh,
It was
nice.
phenomenal. You couldn't taste the vodka at all.
Of course.
And then they had the bourbon out there, and he said it wasn't quite finished. It's like a couple months from being finished. But it was.
So they brought a private stock in?
no. Well, this one they were bringing what they had. And he said it wasn't like he wants it to be.
You
aged.
couldn't buy it there, is what you're saying.
No, you can buy
Okay.
it there, but
Gotcha.
yeah, the stuff that the bourbon that he had was not quite finished, but he wanted to bring it, and it was a couple months from being finished. It's still delicious, and I'm a bourbon fan. I was hoping to get their gin because for me, gin is a new. I have not been into gin because I don't like the juniper berries, the Christmas flavor, that like tasting pine trees, but they focus more on the citrus aspect of gin, and I was like, oh, that'd be really interesting. So he did not have one, unfortunately. But anyway, I bought this there
Spirit
at Spirit World. Alex was
Down
not
on,
there.
what is it? It's
Exarban?
Exarban, yeah,
Yeah.
Exarban Plaza.
About 70th and Center, 68th and Center.
You'll
somewhere
literally run into it if you accidentally turn shortly.
there. Yeah. All right. So back to your question. Sorry that was a
No, love
weird
it.
tangent. All right. So the answer to your question is yes and no. Yes, we can.
Of course it is.
So, like, when it comes to the woolly mammoth, we can do things like that because we have a similar creature to that called the elephant right now. And we can take an elephant and we can take the missing DNA that the woolly mammoth had that the elephant nowadays does not have, and we can create that.
All right, so what I'm wondering is this just like Jurassic Park, where they find the embryo inside of the honeycomb and extract the DNA, and they can create whatever?
Yes.
Is that
Yes.
is that what this is?
The catch with DNA though is it degrades.
Mhm.
Not relatively quickly.
So we have to
Obviously,
fill
can
in the
get
gaps.
woolly mammoth,
Yeah,
but-
and that's what they did in Jurassic Park. They did a good job.
They
of
filled in the
explaining
gaps
that.
with frogs. I recall. I remember
Yeah,
very
exactly, and,
Dino
and other
DNA.
things. Yeah, well, and with the velociraptors, which are too gigantic for actual velociraptors, but there were other things in there.
With CRISPR, it can be as big as you want.
Exactly.
So, my
but then,
wait a minute.
So you're
Wait a
talking.
minute.
so
Wait a
you-
minute.
Yeah.
Retract that statement. What?
Trademark before Viagra trademarks that.
Exactly. It can be as big as you want or as small as you want. Is that what you're saying?
You know,
Like,
whatever.
well, you could adjust
You could-
sizes.
So yeah, you could
No
take
one wants a
we'll make a
small
tiny woolly
velociraptor.
mammoth.
you could take a small dog, and you
Uh-huh.
can make a big dog.
Exactly.
But if you wanted to add a feature that wasn't presently or beyond or on there before, like if you want to add wings to something, let's just say, and this was on the Radio Lab, the when pigs fly. So if you want to add wings to pigs, because of evolution, they didn't have wings. They don't have the structure, the bone structure to do that. So you can't just add wings to a pig.
Like you could add wings, but they wouldn't
So,
work.
Point of correction.
Maybe?
You could add wings, but probably not very functionally.
Yeah.
Right.
Because, again, and this is the whole this will kind of enter us into this conversation about humans too, but we're complex organisms.
Mhm.
And if we're complex, so is a pig.
Sure.
And to try to add wings, you're not only just adding these objects that are wings, but we're also adding in everything else that needs to be there for
Yeah,
those
where
wings to
do you add
function.
them?
Yeah, the
and
weight.
how they
The
move,
weight, the
structure.
muscles,
Right.
yeah, that makes sense. My question about let's go back to the
making
woolly.
fairies.
No, I get it.
Loosely related.
But my question is about the elephant woolly mammoth example,
Mhm.
and specifically, okay, so we have we have a species that's similar, right? And then we have probably the majority, if not all. Do we have all the DNA of a woolly mammoth? Is that like a thing?
I
mean,
know they
or-
accessed it through bones.
Yeah,
I'm not
and-
sure if all of it's accessible or
And we had a pretty
usable.
complete.
Well, so
was it Alaska or northern Canada? They
Stuck
found like
in the ice.
It was a
Stuck
pretty
in the ice.
yeah.
let's
I don't know
let's-
why elephants
let's-
would be
let's-
like this in the ice.
But they're
but-
probably doing that.
let's
upset
pretend
about being in the ice.
Let's make a hypothetical that we have the full sequence of DNA of a woolly mammoth.
Sure.
Okay, and then we have the DNA of an elephant, but we want to create a woolly mammoth. So we try to take the DNA of the elephant and use CRISPR-Cas9 to replace segments of the elephant's DNA with woolly mammoth DNA to recreate the woolly mammoth. Would it be? I'm asking about the degree of accuracy. How accurate would that DNA be to the woolly mammoth, to the actual woolly mammoth DNA? Or would there be discrepancies in little one-offs or little...
How many generations would it
Yeah.
take?
No, that's
to,
a great
to-
question.
to do that.
That's awesome.
Well, so think about it like a scantron. I guess here's the teacher talking about scantrons.
I love it.
Back in the good old days. So you have the answer key, right? This is your elephant.
Mhm.
This is your elephant's genome. You have all the DNA of the elephant sitting there right in front of you. For the most part, you know what all of it does. Maybe you don't know what every single strand, every single gene does. And then you have this other answer key, or the whatever some student turned in, and it's the woolly mammoth. Not quite there yet. There's a mostly correct, but there's some weird instances of
discrepancy.
yeah, a discrepancy. What you do is you align that, figure out where those points are in that genome that they've kind of separated, and then you use that CRISPR and then the Cas9 protein that it creates, and then you have that go and cut out that incorrect part, introduce your own chunk that's the correct chunk into that strand, and you just do that X amount of times, probably thousands.
How long does that thousands of times take in practice?
Well, so you're so
today or in
a day.
I mean, to make it, to,
Okay.
yeah, to actually do it, but to make a viable embryo that would actually become a woolly mammoth,
Sure.
you're talking probably weeks.
Again, it's still like days
Instead
and
of-
weeks. Like you're splitting hairs on days and weeks. We're
I
not
know.
talking about
Now-
centuries here.
Let's so in your case, you said, let's say we have a completely viable sample. Let's also assume then that we've tested what they would likely do is take a chunk out, add it to the embryos, see what happens. Take a chunk out, add it, see what happens. But let's say we've figured out all of these chunks that have already
Okay.
we've determined these chunks all kind of work in some way or another, and we need all of these chunks to create the hair and create the size and create the unique features.
Right, and there, sorry to interrupt you, but there comes the ethics question when it comes to human embryos, right?
Yep,
like trial and
exactly.
error with human embryos is like not okay.
And I have some facts and figures on that. So
Okay,
we'll get to
noted.
that.
But keep going.
But no, you're exactly right. So you have now at this point, assuming you've tested and tried everything, it would only take a day or two to throw the Cas9 in there with the correct sequence that you want in there, and that Cas9's, regardless of anyone's time frame, is going to go in there. It's going to go chop out that DNA you wanted to chop out, and that new sequence you've also kind of thrown towards that general vicinity is naturally going to plug right in.
So within 3 days, you could have a complete project. You're like, yep, got it done.
And then you got to wait for it to grow from an embryo.
Yeah, because I mean, like you're going to do this amongst a lot of embryos, and then which ones are going to be viable. That's going to take a little bit of time to figure out which ones are going to be viable.
What
Right?
I always, no, that's exactly right.
Yeah.
What I don't know. I feel like I'm always optimistic on the time frames of these things, because all it takes is 100 labs to work together on a woolly mammoth, and they could all be trying one embryo or one chunk of DNA at a time.
Mm-hmm.
And then as you add all that up, that time frame of
It's
creating
way
this master
shorter.
woolly mammoth
Yeah.
goes down significantly. So
Well, and that's I mean, I kind of get into ethics because I mean
Yeah.
we're starting to get into that. a
Yeah,
little
we're getting
bit.
there for sure.
So I
fine.
do want to start a little bit softer right now with things that and what was it? You had notes on these on the therapy versus enhancements.
exactly.
So in that video on Netflix, they talked about therapy and enhancement. Enhancement is instead of having brown eyes, you're going to have blue eyes, or instead of
Oh,
having
yeah, we kind of talked about
a
this.
yeah,
Video, we kind of
instead of a
you're
skinny frame, you're
snipping it
muscular.
off and, yeah.
So
replacing
that would be enhancement. Therapy would be more of you had this disease.
Muscular dystrophy.
Yeah, we're going to get rid of that disease for you.
I don't know, man. Some of those are really black and white, and some of them have like a really gray area.
Exactly.
between what's what.
And what was interesting, I saw, and
I'm going to cut the fat gene out.
well,
Yeah.
You're not going to be
Yeah.
fat anymore. It's like, whoa.
we kind of
that's-
need fat, but not excessive.
Right.
Now, so other things like I'm going to mention two here that were in that Netflix program was dwarfism and people that are deaf. Now, for
And Down syndrome, I assume.
well, okay,
That's been
that's
discussed.
another that's another topic I want to get. Yeah,
Yeah.
let me get these two because that's already being discussed in Iceland.
Okay.
and all
But
that.
the world needs those people.
so that
You know,
so here's
kind
the
of
thing,
diversity.
like for dwarfism. I mean, I and I don't want to say most people, but there's a lot of
The
people.
majority of them want that. They're like,
Well,
I'm cool with the way I was built.
being
and
a
made.
dwarf, yeah,
Yeah.
actual people with dwarfism are like, well, this isn't
Yeah.
an issue for
You
me.
guys see it a problem. I don't see it a problem.
Yeah, the problem is how you treat us, but not how we are. We're fine.
Yeah.
So, do we eradicate that? Where's that line? People that are deaf, is that a disease? Can we cure that? We can certainly cure it, but the people that are deaf are like, well, it's not really a disease for us. It's just it's a way of life.
a
and it's fine.
different way of living. Yeah.
Yeah.
Especially when that situation, whatever you want to call it, turns into a community. When there's a community of deaf people and a community of people with dwarfism, right, that becomes a community and a culture. And now you're not eliminating in a physical attribute, you're eliminating a culture and a group of people, which is different, right? That's a different thing
Right.
than eliminating a physical attribute.
Exactly. And then we get into Down syndrome, where places like Iceland have almost eradicated Down syndrome using abortion. I mean, they've been done pre-screening, and they're getting rid of Down syndrome by aborting fetuses that have the gene.
Almost eugenics.
Yeah, basically.
Really? So they're testing the pregnant women and going, it's not
Oh,
going to
we-
be a good one.
Yep.
We do that here.
Yeah, yeah. I mean, you do pre-screening for if you go to hospitals and get all that.
You
Okay,
can.
yeah, I
You don't
guess.
have to.
Yeah,
but they,
exactly, you can.
You can.
Yeah,
No. I was going to say, I never did. But
yeah.
I was
It
like,
is an
I'm gonna
option.
have a healthy
yeah.
child, like.
I would say most people would do it, but yeah, it is an option to do that.
And again, most people with that current technology, assuming or kind of ignoring CRISPR for now, most people are going to take that option just because they want to prepare themselves. What do I need? What do I need to know if my son or daughter is going to be predisposed to Down syndrome or predisposed to or MD? You want to be able to be prepared for those things. So, kind of in that innocent boat, it makes sense to do a screening because you can.
It's crazy
theoretically-
because, just, 20, 30 years ago, it was like, we're having a child.
Yep.
Okay, here it comes.
Yeah,
You
Doing
know?
it.
look, boy,
Hopefully
girl,
we're
we
not-
don't know.
We don't know.
Yeah.
We don't care. Let's bring it
We're
on.
coming up feet first, head first. I don't know. We're gonna figure
But
it
in
out.
the last-
Get the salad spoons.
Yeah.
In the last 10 years, I mean, really,
right.
It's a huge jump. And
it is a
it's-
huge jump.
And that
I guess
jump-
I didn't even know it was an option when I was having a child. I was just like, hey, cool. We did the whole, mammogram thing, and it was like, oh, we're having a boy. Cool, I didn't know that I had the option of checking out whether he was
Screening.
Yeah, I didn't know about that type of screening.
No, those screenings
I guess
involve
no one
like
offered
a,
it to me. But
Those screenings do involve like a syringe into the
They do,
into
yep.
the embryo, right?
Right into it.
Of
to
the
test
child.
The DNA.
Yeah, to pull the
Yep.
DNA out and test the DNA. So that's happening. I mean, they did that with R2 pregnancies.
And one
Really?
thing you always want to ask yourself too, which is kind of a challenge, is what was it like before ultrasound? You know, I'm sure there was a group of people that said, "Don't get an ultrasound. You need to just have the kid like we did, and everyone else did before you.
you
The
know, it'll be
ultrasound's
fine.
not going to help
Yeah.
you. You're going to be fine."
Yeah.
But when you're asked that as a parent, you're going to say, "Well, of course I'm going to get an ultrasound. I want to know how my child's doing."
Yeah.
And kind of in the same vein, if someone could say, "Hey, we can help you prepare for any problems that your son or daughter might have,"
Right. You
They've
want to make sure
almost
that it
put
got
that-
that beating heart
Yeah.
still, and you know.
who's going to say no to that?
Right.
I mean, you can make an argument for it, but for the most part, you want to do the best for your child. So the pull is always going to be towards, well, I probably should morally
But that issue of
be there for
But
him.
that issue of, as a parent, you want to do the best for your child. So then that begs now, as technology has progressed,
CRISPR.
that now begs the question. I want to do what's best for my child, so I want them to have no fat and be super strong and have blue eyes.
Better,
and,
faster, stronger.
And oh my goodness!
Well, and that's
right?
the thing is we're not that far.
So-
We're truly not that far away from some cures for significant diseases. The problem again with humans is not everything is a one gene, one and done solution.
Sure.
You can't just cut out a cancer gene and be cured of cancer. Cancer is a very complex system of several different genes interacting with one another.
But some are right.
But
Down
there
syndrome
are
is-
some
is
things that are
one-
relatively simple.
It's one.
Down syndrome is complex, but yeah, it's
23rd
kind of
chromosome,
the additional chromosome.
Okay.
Some things are just like one letter.
But
is
that's
off.
exactly. Some things are very simple to fix that we could theoretically, like MS or
Yeah.
muscular dystrophy.
And that's
I'm
the
sorry.
ACTG that we talked about with
Yep.
DNA strands. That it could be one letter off.
Yeah.
But
Cure that, please. Let's do that.
and it's
I'm
it's
on
going
board
to
for
happen
that.
within,
Right, okay.
I would guess, 5 years.
Right.
They're
and-
going to have a cure, a CRISPR cure for some disease. Let's say Alzheimer's, for example.
And this is happening very quickly.
Right.
Like you said, years.
they're saying
not
10 to 20
decades.
years. I'm saying 5 to 10, and
Yeah.
that's giving it a lot of time.
Just because it's so inexpensive and so many people
It's
are
like
doing
the
it.
internet. I mean, we've now discovered this system of data, and now we haven't even begun to utilize that data or compress it or compile it or really test it.
Right.
And we have a lot of people have no knowledge of where that can go. But as a kind of a kid of that internet era, you realize pretty quickly it's not
Oh,
going
yeah.
to take long.
Oh yeah.
People are going to figure it out pretty quick. And that, again, gosh, you can go into 8 different directions with this. But you can see kind of how hard it is to legislate that. Because let's say the United States says, nope, we're not going to do it. We're going to be very strict and controlled. It takes one other country to say, well, we're all going to have super high intelligence and crazy strong muscles, and
We win.
we win.
Yeah.
And we're never going to die, so
that's-
we're done.
That gets into designer babies. I did want to say one thing on the disease portion
Sure.
before we start getting into designer babies. And
I just want to be
designer
mouthy for 1 second because
Okay.
I'm like 3 beers deep, which is that especially with our Congress and our House and Senate, because you've got these old dudes and ladies that have no idea, who are not children of the internet generation, trying to make this legislation. They have no idea what they're
Well, you watch
talking
the
about.
Facebook.
Yeah.
Yeah.
You watch the
Not-
Zuckerberg.
Did you say
Right.
the Facebook?
The
The-
Facebook
That makes you
the-
old, right?
conversation.
the Facebook
Back
conversation.
in my day.
We got you.
The
Um,
cyber. That was
so
a Hillary
what-
Clinton quote was yeah, when we're talking about the cyber.
Yeah.
Oh.
And wah, wah, wah.
What I wanted to speak
Was that
to.
all a sigh of?
Yeah.
Oh,
Yeah.
for one for her, like
Yeah.
oh.
Yeah.
I wanted to speak directly to the disease portion when we start actually curing, being able to cure certain diseases and all that. We go down that rabbit hole and we don't know where the line is, and ethics are still a part of that. It is going to be, it's going to become in our society actually unethical to not use CRISPR.
100% agree.
Yep.
100% agree. You would be, and this will become the standard is you would be a bad parent
To not use
to
it.
not do, because let's say you go there,
Why
and
would
you
you-
go
why
through
would
your
you want-
ultrasound,
Yeah.
and you go through this probably commonplace genetic testing, and they say, hey, your kid is very likely going to die at childbirth because of this disease. However, we can just insert CRISPR, fix that disease, and your child will live.
Mhm.
That would actually be,
Who's going to say no to that? Who's going to
Right.
say, well, that's okay, my child can
no,
die.
I'd rather
Yeah.
have that child live in an incubator for the first 8 months of his life.
But
yeah.
okay.
You know
True.
what I'm saying?
No, yeah.
There's
what about
there's-
Alzheimer's? What if they're predisposed to Alzheimer's?
Well, okay, so part of this, and
cancer.
I don't want to get into this podcast because I do want this to be like a part 2, but I think that the what you're talking about, like a dies at childbirth, but we can fix that.
Yeah.
That starts to get into like the faith.
Sure.
Your faith is going to dictate of whether you do that or not, I think. And I didn't want to bring that into it here, but it is a portion of this, and I do want to.
I think it
I
has,
want to
I
do
think
it.
it has a
Absolutely.
significant place
Yeah.
in this conversation.
But I want to do a part 2 specifically on that one.
You-
on
good call.
faith.
You're
Yeah.
you're literally playing God.
Yep.
Splicing into human genes.
Mhm.
Splicing into corn or things we eat.
But that's still. I mean, that's
That
still
also is playing God or
Yeah,
creating a
it-
species.
It
yeah,
is.
It's just it's ethically okay because
you're
well,
making
it's
it better
corn.
to.
It's not
you know.
sentient, at least.
Yeah,
Right,
exactly.
right.
That we know of. You know,
And I was
It's
just
not,
naming
Brian,
that one
it's
thing.
not.
And-
not part of the tree life.
but yeah, I mean, you're literally playing God.
Yep.
Is what you're doing.
And we've been doing it for 100 years,
Well, yeah, because when we talked about GMOs, I mean,
200 years. maybe,
what's your version of GMO? I mean, are you is it using CRISPR to actually splice DNA, or is it selectively breeding plants?
right? Mendel,
Yeah,
Yeah.
because we've been doing that for
plenty of years.
millennia.
Mhm.
I mean, thousands of years we've been doing
This
that.
corn grows better here. I'll plant it. That's GMO, right? Like technically.
right.
Yeah, I mean, where's that line of GMO fall?
Or
for you?
combining different species.
Yeah, exactly. Yeah, I because of this climate, I need to add this with this,
Right.
and
Yep.
it works. And that's not going in gene editing. Oh, you guys are on to the next one. The Dark Paradise, is that next?
Talk too much, Brian.
I
It's
know
a
that
good timing for the.
we've
good timing
got
for
somebody
Dark
over
Paradise.
here
The
that's
Dark
only got
Paradise,
about 15
yeah.
minutes, and then he's gonna zip out of here.
Oh yeah, that's right. Yeah.
I got a short clock, folks.
So this again is from Kros-Strain, and it is it's called Dark Paradise. I think you can extrapolate why I chose
Dark Paradise, man.
the name of it.
That is dead
because
on.
we're start getting into the Dark Paradise.
That's
of this.
creepy.
Yeah.
You mentioned the Radio Lab podcast. I loved how. Appenrod is one of the guys, and the other guy.
I don't know the
I
names.
can never remember. But the entire story that they're doing of this CRISPR story, he's saying, "Well, come on, guys, though, what's going to happen? There's a
Right.
dark road this can go down. There's an ominous cloud coming with this technology."
Right, absolutely. But that being said, I want to talk about this beer real quick, and then we'll get into the last bit. It's, again, Kros-Strain, Dark Paradise. This is like an amber ale or a red ale from Kros-Strain. It is 4.3% ABV.
Oh, nice, actually light one.
Yeah,
huh?
a little lighter. Lighter alcohol, darker beer. So,
I was going to say this is not lighter
yeah,
in color.
lighter in
It's not light
alcohol.
beer.
lighter in alcohol
Yeah.
content.
I still have to finish mine, but
Yep, I got
you
you.
guys can go ahead and.
I got you here, bro.
Oh, you got
You know, CRISPR could help you chug that faster. So
That's interesting.
the
Like,
wins are never ending.
really? Like, could you alter a DNA segment to make it so that our blood is less
Hemophilic?
susceptible
Like-
to?
So there are several scientists. I'm going to go completely off the deep end on this one.
Go
Do it.
ahead.
Go
Yeah.
for it.
There are several
We
scientists.
never do that here.
And by several, I mean hundreds. Yeah, right. Who are trying to get death put on an illness list rather than on
Mhm.
Whoa.
as
I've
death.
heard- I've heard
Slow
about
as
down
in,
here. Slow
as
down.
in
Yeah, I've heard
They're
this
trying to
before.
get
This is
death
a-
put on what?
an illness list. Death is not this end all. This
It's
is
an
just
ill.
part of a
If you think about it,
issue you have.
if you think about it, in this case, Brett,
Crazy, right? I mean, but it's
Death and
mind-blowingly-
taxes.
Yeah.
Death and taxes. Like, that's what
You
it
can
is.
fix one of those, and you're
Yeah.
just stuck with taxes forever.
de-
Who doesn't want that life? Taxes forever.
Awesome.
think about life as actually curing death. We can
You
cure,
fix one, and you're stuck with a
Yeah.
lot of
Lots of
taxes
taxes.
forever.
When I hit about 90, if I hit 90, I'm going to go.
Now here's,
no,
I'm not talking
I'm
90-year-old you. I'm
done.
talking this-year-old you
I'm done.
for 300-400
Oh,
years.
well, yeah. I mean, no, well,
Not so
maybe
shabby.
10 years ago. For
Yeah.
me.
You know,
there you go. You
keep
pick,
that
you
forever.
pick.
Brett, you know what the scientific community has already said. The first person to live to be 150 years old has already been born.
I've heard
Oh.
300 or 400.
I haven't heard that yet.
But again,
Oh, yeah. I
you're
mean,
this
with
tapping
technology is
modern
gonna-
medicine, yeah. I mean, I guess, yeah.
Yep.
So, well, let's cheers on that.
Yeah,
To 300.
To 300.
like not Sparta
Think
300.
of how
Right.
many new beers you can try.
You know, you
and not get drunk while you drink them.
Oh, and stay thin while drinking this.
Exactly.
this is a
Ooh.
nutty. It's those like I get
Ooh, I
nutty.
can taste that. Yep.
Yeah, it's good. Smooth. It's not assertive, but it is dark, so it is heavy flavor.
I wouldn't say coffee. It's not coffee.
It's not quite
but
coffee.
it is nutty.
There's like a hint, like if you it's not
like
coffee flavor,
like a
but there's
rich mahogany.
No,
But I wouldn't eat mahogany. I mean,
No, it's
but I'd smoke
You'd
mahogany.
Smoke or smell.
Yeah.
it's somewhere between smoked almonds or walnuts and coffee.
And this is
It's somewhere in
also
between
by
those.
Kros-Strain.
I do get coffee notes,
Yeah.
and
Yeah,
it
there's a
just
little bit of
hint,
coffee note
just
in
a
there, yeah.
hint.
I feel like these types of beer are hard to produce too because you end up generally getting this huge like
I can't
blast right in your face.
I
and
can't
it's
believe
hard
this is a
to-
4.3. You know,
It's very light alcohol
liner
content.
25 IBU.
It's in the Bud Light category for alcohol content.
Huh.
America.
Right.
Not too shabby though.
Yeah, but actually has flavor.
That's very tasty.
Yeah. All right, Designer Babies, Designer DNA. This is the final ethics portion of the podcast here, and you got
Yeah, here we go.
13 minutes, Mike.
We got 13 minutes.
All right,
I'm going to hijack it with all
so-
technology, just so you know.
Yeah, do it.
so China has already experimented on human embryos, and
Non-viable.
not non-viable. These were ones that were going to be
The
discarded
rules and
already.
regulations over there are a lot
Okay.
different than over here.
Now,
A little bit.
that being
Little
said, when they did it,
Go ahead.
less than a quarter of the embryos
Sorry.
embryos were actually viable. So it didn't work as well as they had hoped.
Okay.
I think they started with 86 embryos and they ended with like 28. That's if my math. These were ones that were going to be discarded
Okay, all
anyway,
right, fair, all right. Try it. Well,
and they were not.
they were going to get discarded because of?
They were going to get discarded anyway. So they were given to science, I'm assuming, and they were never going to actually let them become viable.
To be fair, so now we kind of assume, a lot of people assume that all of these gametes, the sex cells, have to come immediately from a gamete. And nowadays, scientists have found a method over the last 10 years to kind of backpedal a typical cell. So you could take like a skin cell and then backpedal it back to its sex cell form, where you kind of get that broad ability of, I can be anything. I can be a skin cell or an eye cell or
Stem cells
Yeah.
can be
so you can kind
whatever.
of create these stem cells without having to
That's interesting.
I had no idea.
without having to take it from a viable
That
embryo,
is
Yeah,
super
I-
interesting.
which is huge.
I did not know that either. I thought like stem cells, like you had to do the cord blood and get
Yeah,
the stem cells.
not
and-
anymore.
Huh. Interesting.
Man, bring this guy back.
But again, so you think that's a good thing, but then you're all of a sudden you're like, well, now we have infinite stem cells to use for whatever we want.
Nice. Nice.
Is that bad?
I mean, I would like a T-Rex and a woolly mammoth so-
Oh.
in a beer that
just
smells of rich mahogany.
Just a tiny T-Rex. Though.
Yeah.
Just,
Can
you know,
we-
just a
yeah.
tiny one.
Can we create that
Like
law really
one-
quickly?
yeah,
one
just,
that eats
limits
mosquitoes.
the size.
He gnaws on my toes
Yeah.
a little bit.
It'd be
It
adorable.
doesn't
It'd
hurt
be
though.
kinda cute.
Would
know,
you-
It's like, oh,
Oh.
It's like a toenail. It's
And
like
you
a
just kick it, you're
Flintstones.
like, get
and
outta
toenail
here.
clippers.
Would you rather fight 100 duck-sized horses or one horse-sized
CRISPR can answer that for you.
Ooh, nice. I like that.
Now, the
saying.
thing with those T-Rexes, would
Reddit.
they be
That's a
house
Reddit
trained?
question.
one horse-sized
Yeah.
duck.
Well, CRISPR could hopefully house train them too.
Yeah, have pre-house trained dogs.
Brilliant.
I mean,
Yeah, that speak English.
Yeah.
right? Well, only here in America. I mean, if you
Yeah,
if you had a
naturally,
dog,
naturally. That only makes sense, Brian.
yeah, exactly. All right, so let's talk about good bads with the
So,
ethics on DNA.
I'm going to hijack the ethics
Yep.
conversation because I'm
got
going to
10
leave
minutes.
in.
Yeah,
I'm
10
going to leave in
minutes.
10 minutes.
That's fine.
So I want to bring up Ancestry.com and 23andMe.
Ooh. Have
Right.
you done that?
No,
I
the,
have-
the
I haven't
DNA.
done that.
Yeah.
No.
So
I have not.
I have a feeling
not
you're
giving my
not going
blood
to
out.
want to after this conversation.
Well, at the moment, there's actually a YouTube channel called Smarter Every Day, which
love that channel.
I highly encourage. Total free PR for that guy, Destin. If you're there, come join us on the podcast someday. Great channel. But he wanted his YouTube channel is sponsored, and 23andMe offered to sponsor one of his episodes and do kind of an advertisement during his YouTube episode that week. And he originally was going to decline because he was like, "Man, I don't know if I want the association. I don't know if I want the risk. You know, he just didn't know how he felt about 23andMe." So
I don't know much about 23andMe. I know
I do. You send out your DNA and they'll tell you
Oh.
where your ancestors
I didn't know that's
from.
what it was called.
Yeah,
'Cause
yeah.
I, okay.
23andMe.
right.
All right.
So
Your
there's a
chromosomes.
couple different services that do that. Ancestry.com is one.
Yeah, that's
23andMe
what we've gone through.
is the other
Ancestry.
one, the major one.
Okay.
And so he said, you know what? This guy, he's great. Great YouTube channel. He says, you know what? I will gladly accept your sponsorship if you allow me to do one episode on your service, specifically on the security of the privacy of the individuals.
And this could go good or bad
And
for
it can
you.
go good or bad.
Yeah.
And you know what they said? 23andMe said, bring it.
Nice.
And he went and he interviewed people, and they let him into their labs, and they let him into their facilities, and they looked at their destruction processes and their privacy and everything. And here's what I discovered. So I'm the cybersecurity privacy guy, right? So what they discovered was that 23andMe is actually doing a great job. When you do your identification, you swab your DNA with like basically a Q-tip on the inside of your cheek, or you spit into a test tube, and then you seal it. And then from that point on, everything about that entire process is nothing but a barcode and a number. Your identification, your identity about that UPC,
You're just a barcode and number.
you're just a barcode and a number,
Yeah.
and like an address for them to send you the results, which is that it could be a privacy issue,
Yeah, a little bit of a tie in there.
right? There's
But,
a little bit of time, but
How do you get past that?
but
We're
generally
just going to
speaking,
send it randomly.
but
where?
generally speaking, no one at that organization
There's
knows your
one team of
name or
people,
anything about
that's
it.
it.
Yeah, and then
the barcode gets sent to those people.
And then
That's
all
it.
of the results, like if you send in your results and then you like whatever, once they're done, there's an option to have them store it or not store it. And if you choose don't store it, they literally put it in an incinerator, and that stuff is gone forever.
Wow.
So they really manage the privacy aspect well.
But if
here's
they
the-
store it, then
But
you can be
here's the-
found, through Ancestry.com.
Through this is 23andMe.
or
I don't know.
23andMe.
I don't know about the security and privacy aspects.
But they could
of
be like,
Ancestry.com.
hey, you have this ancestor in California.
Well,
You
here's the
don't
thing.
know.
Knowing Ancestry.com, think about what Ancestry.com has about you as a website.
Oh, yeah.
So if you go onto that website and you fill out all that stuff about your ancestors
Yeah,
and
social
your
security
siblings
numbers
and everything about you,
Mhm.
and then you do the DNA thing, I don't know how they segregate those two things. Maybe they do. Maybe they do it well. I don't really know.
Interesting.
But those two services aside, right, free pub for them right now. I don't know if you should do it or not. There's a ton of different aspects there, but let's look at the broader picture of having organizations storing
Your data.
DNA, your
yeah.
genome,
Yeah.
right, basically, in a system. Let's pretend that one of those two services does a terrible job of privacy and security, and they've got your entire genome associated with your name, address, social security number, and everything else in some database somewhere, and then that gets hacked.
Well, no, this is starting to sound like if they have that, DNA and all that data on you, they could put you in a category. Brian, you and I talked about this, about social status.
Correct. You're by nature establishing basically a caste system. Imagine
Like
how,
you're prone to
Imagine-
having a disease of this nature, so we're not going to
Yeah.
allow you to do whatever you
imagine
need to do.
how much an insurance company wants that information. A health insurance company,
Yeah.
right?
And they're going to get it.
Absolutely.
And they're going to get it.
So I have a quick question, just a little interjection in here.
Mhm.
So you're talking into the future here, and in the future, what if
Ooh,
it?
not in the future? It's,
It's
happening
so
right now.
inevitable, though.
I'm not saying far
Mhm.
into the future. I'm just saying in the future here. What if it becomes so commonplace that instead of like you have to provide your name, address, social security number, fingerprint, DNA, blah 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 blah, they take your DNA and that becomes your identification.
So
And it becomes so commonplace that they don't need to store it anywhere. They can just once they retrieve it, we have systems in place that it
Your
becomes-
DNA is everywhere.
So they don't even need your DNA. So the Golden State Killer that they caught, did you guys read up on that?
Yeah.
No, tell us a little bit about it, because
So-
the general public might.
Was that
Golden
the
State
was that the
Killer.
rapist?
Yep. And
Okay.
Golden State rapist. 30 years that he couldn't go. That they couldn't find him. They had his DNA, but that didn't do much because if they don't have his DNA, like in a database somewhere, it doesn't matter. And he obviously wasn't frequenting 23andMe or Ancestry. But the cool thing about DNA, unless you're the Golden State Killer, is that it's pretty relatable to your relatives. All it takes is one relative to also go on that site. And then they, I believe, went through 23andMe, the police department in Golden State in San Francisco, and asked for just a comparison from their DNA that they found at a crime scene to anyone on their database. They didn't find the Golden State Killer, but they found someone related to him.
Mm-hmm.
Wow.
Because you can match that DNA up. And all of a sudden, they don't even need your DNA. And it's kind of... And that's how they caught him. So, you know, theoretically, that's a good thing. You caught a serial killer and a serial
nice
rapist.
job.
Kind
Right.
of,
But
Extrapolate
what
that,
happens
right?
on the other end of that?
Yeah, where does
you
that
can
line-
track relatives down now? It doesn't even have to be you. And I know a kind of a related story is the Facebook infringement issue that they had. You didn't even have to say, my wife is huge into privacy on her Facebook, and she was always the one telling me, you need to fix this on Facebook to ensure that no one sees this.
I'd be glad to have that conversation with you.
Oh, yeah, totally.
Have you seen the bad lip reading on that yet?
It's awesome.
Hilarious.
So, of course, Facebook releases the people who got hacked, and I didn't get hacked somehow. And everything's global on my account. And she got hacked, and she's like the privacy whiz. And all it took was one friend of hers to have open rights,
Yep.
which was probably me. And anyone could see her profile from that other person's profile and gather all of that data. And it's similar to this DNA. It doesn't even have to be you. You don't have to give permission. Related from a standpoint, from a heredity standpoint, that they can kind of piece the puzzle together from that. So they could realize, an insurance company, for example, could realize, well, even if you didn't give me your DNA, I know your dad and I know your kid
Yeah.
both have predispositions for this. You probably are going to have that as well.
Right.
Right.
And
I
don't
know
have a
you got to
choice.
get going, Mike, so I want to give you just the floor for just a couple minutes to vent. and
You know, that was the majority of it. Is that first of all, whenever data is stored in an aggregate system anywhere, it's going to get hacked.
Mhm.
Just consider that data hacked. Facebook, done. Right? You aggregated data, it's done. The Department of Defense Office of Personnel Management, done. Right? That got hacked. Like your semblance
Click
of
icon and hit 11111.
yeah, all semblance of privacy is out the window. Like using a social security number as a unique identifier for humans now is futile, right? It's done. Everyone knows your social security number. It's out there. So knowing that and that large data are going to get breached, considering the notion that there are several 3-letter acronym organizations and corporations that would love to do away with putting a driver's license number on your driver's license and just do unique identification through your actual unique identifier, which is your DNA, right? We're just going to grab an eyelash or a skin sample or a tissue rub on the inside of your cheek or a tissue swab
Or a relative.
or a relative.
You're not
and
gonna say
we
yes,
could just call it
we'll find it.
Gattaca.
we don't,
Yeah.
we don't,
I mean,
we
like,
don't
seriously,
need to
go
do.
go watch that movie.
You don't.
Yeah.
You don't,
If you haven't
you
watched
don't
it,
need
yeah.
a primary key in any database any longer. It's your DNA, right?
Right.
That's your unique identifier, and it knows every single thing about you.
And it's not hard to get.
No.
Right.
But
And
that's something you can't fake.
You can't fake it, and you
Well,
can't. You
in
can't change
Gattaca
it.
you kind of did, but you know,
Well, he tried.
Up
You
to a
tried.
certain point.
yeah.
CRISPR could fake
You
it. I
can't.
mean, you know,
you
throw
can't
enough
fake it, and
CRISPR,
you
yeah.
can't change it, right? I can't go out and make my Facebook profile. I can go out on Facebook and I can make my life look like the greatest thing that's the greatest life that anyone's ever lived.
You post everything positive, nothing negative.
That's what your Facebook is, right?
Well, yeah, my sunshine, rainbows, unicorns, glitter all the time, but.
And fairies.
And fairies. But yeah, with your DNA, no, it tells the whole truth, right? It tells every single
Nothing but
physical
the
attribute
truth.
about you, and you can't get away from it.
Yeah.
So
What you're predisposed to and all those factors of your life and who
at
you are.
some point, I look at Moore's law because I look at technology and the rate at which it accelerates. But we'll have the human genome mapped to an extent, and I don't know the time frame. You can help me out on the time frame, but the time frame irrelevant is. At some point, they're going to be able to take maybe not even your own DNA, based on what he's just said is, they're going to take your dad's DNA,
Yeah.
your mother and father, or your father and son, or your father and brother, and they're going to put that together, and they'll just pull your image up on the screen and be like, well, that's you. We know that's you. I don't need to. I don't
Who's your
need.
mother. Who's your father?
I don't need the.
And we know your DNA,
I don't
we
need
know
the
what it
police department
produces.
artist to recreate a sketch of
Right.
your image based on the description from the witness. No. We know what you look like. It's right there.
And even if we don't know what you look like, we know what your DNA sequence.
Right.
We have that.
And we'll find you.
Yeah.
Like the next time you sneeze, we're going to find you.
Another
Exactly.
snippet of Science Hope. epigenetics will help fix that. And that's a whole other episode.
Yeah.
What is that? What does that mean?
Whoa.
Just
Just
the best thing.
Tell
ever.
us about that.
Touch on it
Second
for
best thing
give it 30 seconds.
A
So we always
synopsis.
had this nature versus nurture, and most scientists for a long time always said, well, it's nature, right? You have your DNA, that's what you get. You're not changing it. You got 50-50 from your parents, and that's who you are. Epigenetics says, well, there's this methylation we can turn on and off some of our genes that get passed on based on the upbringing you have. If you're in a stressful environment, you're more likely or you're more predisposed to these things because your DNA changes its form in response to those stressful environments. So theoretically, you may not have that exact copy of your parents because of this epigenetics, this reaction to the environment you're in, which plays a significant impact on what you look like later on. So,
I know
Right.
this is great stuff.
Man.
Really, we're gonna end this in like 2 minutes.
We're gonna end it.
Well, because
He's gonna cut out, but we haven't solved nothing about it yet. We can keep this going.
Okay.
If you got to dip out, you got to dip out.
I do. I have to
You-
tap out.
You gotta
I'm going to finish my
finish your beer.
two sips of beer here.
Let's
Yeah.
just keep it going. It's
So
no big deal.
the-
It's
the
great conversation.
The thing that we were leading up to was the designer babies and designer DNA. So some of the problems with designer babies is like we can't. Do we have the technology to do that right now? Potentially.
Sort of, yeah.
Well,
Potentially.
Chinese are already starting
Okay.
to try to do it.
But it's
Is
not,
that what you were saying?
It's not proven technology and it's not reliable technology yet. But we do have the technology to do that.
it's a matter of time,
Right. It's just a matter of time.
and not a whole lot of.
It's
But then-
it's trial and error,
gonna
and
happen,
it has
you
to
have
go
enough
down.
trials, you're going to eliminate
Exactly.
the errors.
Where does consent fall in that?
That's a huge thing.
Well, and with China, they're trying to find you're saying embryos. Well, those that want to abort their babies, instead of deporting them, let's
Aborting.
deport. Why did I say that?
Deport.
Sort of deporting them from the.
That is, it is a way of
Deporting
deporting.
them.
aggressively deporting.
But
someone.
instead of just letting them actually go away, let's use them for science. Let's try something else, and maybe something awesome
Right.
happens because of it.
So you bring up a good point, which is consent.
Mhm.
Do you? Okay, it's personal questions, sort of loosely personal. Do you
Mhm.
vaccinate your kids?
Yes.
Okay. Do you ask for their consent when you vaccinate them?
No.
Okay, but you do it because why?
Because of the potential harmful effects. The other side.
Because
Totally.
says that I have to
Right. Yeah.
That too.
Oh,
That-
I mean,
that
Schools
too.
do say it,
That's
and
okay. There is
rightfully
yeah,
so because
there's
we don't want
waivers,
want
but-
flu to outbreak again.
Absolutely. We don't want the
Can I-
whatever virus that
Or-
was
I want
in
to. I want to throw out there.
1914.
Do you post pictures of your children on social media?
Yes.
I very rarely do, but other people do. So
Yeah.
this, like my kids' generation, is going to be the first generation to grow up 100% on social media.
Yeah.
They're
Yes.
already aware of how awful that is. This is why Snapchat became a thing.
Yep.
And that's
And
why I don't use
immediately there and
Snapchat.
the average age of a Facebook user 10 years ago was like 15 to 30, and now the average age of a Facebook user is 25 to 55.
Oh,
It's
and
getting
There
I
are
would
no
say-
kids on Facebook
quickly.
anymore.
I would
no,
say
they
half
don't.
of my news feed is people saying, "Hey, do you remember this?" And it's something from like barely my childhood. It's older than my childhood. I'm like, well, I mean, I kind of remember that as a artifact of my childhood,
Sure.
but that's half my news feed. Like,
Yeah.
oh, click like or share if you remember, if you know what this is, or
Right.
you know how to use it.
Yep.
So yeah, it's getting older, the social media aspect of it. Something else with designer babies, something interesting that I didn't know before this, like if we add CRISPR to you right now,
Sure.
that doesn't really affect your offspring as much.
Not necessarily. No.
If you do it in an embryo, that affects all the it's a permanent change in that embryo, and it affects all offspring
and that's
of that
the
embryo.
big kind of legislative catch that they have right now. So you have somatic cells,
Mhm.
which that's your eye cell or your skin cell. It's already decided what it's going to be, and throughout the course of your life, it's always going to be an eye cell. It'll reproduce. It'll have
Mhm.
all of your DNA in that eye cell, but only some of it is going to express itself. It's going to express the important eye components of that gene or
Right.
that set
So-
of genes.
So wait, how does that impact what we talked about earlier with like curing diseases and such?
Now, maybe I want to cure macular degeneration. I'm not going to necessarily target a gamete in a patient, I'm going to target their eye cells.
Depends on if we
specifically,
want
and
to put.
That's not going to be
yeah.
passed on to a.
It depends on if we want to put CRISPR on autopilot for that, or are we doing this in individual people?
And now, even if you put it on autopilot, if you're putting it in a somatic cell, if you're putting it in an eye cell, that eye cell is never going to go on to your
Kids.
kids.
Because
You're targeting specifically eye cells, which don't.
And at that point, you've
exit
probably
that way.
already had kids, so
Right.
you're not passing on your genome anyway.
Right. So, but if you target gametes for, say, muscular dystrophy or ALS, then all of a sudden you're targeting a
Human embryo.
reproducible embryo that can be any cell and will get passed on.
Yeah. So what we're talking about, Britt, is
No, I'm with
yeah,
you.
yeah. At the adult stage,
The
if you,
door is thin in the bathroom. I can hear what's going
Okay.
on.
Gents, I got to bail out, okay?
Mike,
I appreciate the evening.
that was terrible.
Do it again. Let's do it again. Let's do it again. No, no.
CRISPR can make you smoother.
We'll see you guys later.
Mike, it was good to see
Yeah,
you.
it's always good to see you, man.
Yeah, thank you for coming on the program.
Meet you.
Yeah.
I'll see you guys
When
later.
you get home, you can still watch us.
I will. I'll jump on when I get home.
There you
Nice.
go.
All right, perfect.
Ask questions.
All right. So yeah, that's something that to be said. You know, you combine consent with the fact that if you do it on a human embryo, that's a permanent change that affects every generation after you. And man, that gets the ethics of that are
Right.
mind-boggling.
It totally is. But then you start, if you ask yourself, what other permanent changes do we kind of force on people?
Mhm.
And there are a significant amount of them that we're
True. That's very true.
like morally
And
okay with.
where's that line?
Oh, totally.
That's where ethics comes into this. Where is that line?
Look at from a religious standpoint, circumcision is required by some religions,
Mm-hmm.
and it doesn't necessarily have a significant health benefit, but we require it
Right.
in those religions.
Yeah.
And there's no
I don't know. I
consent
wouldn't say
involved.
it's a significant health benefit, but I mean, it is cleaner,
Yeah.
I guess. You know?
Yeah, I mean, you could. But if I could say you get to choose between a circumcision or curing muscular dystrophy in your kid, you're probably
Yeah.
going to pick.
Yeah, and then
Muscular dystrophy.
we talked
Right.
about
I mean, theoretically, you know.
and we
Sure,
talked about
sure.
that before of the ethics of at some point it's going to be unethical to not use CRISPR.
Exactly. Because, what parent is going to say,
Oh, I
no,
want my child to
I
have
want my
this."
child to risk it?
Yeah.
It's one thing to kind of avoid something like a GMO organism. If I want to not eat GMOs, it's pretty easy to do. It's not going to affect me too much.
Yes and no. I mean,
It's
difficult
to avoid
it's
it.
difficult
For
to avoid
sure.
it nowadays.
Right.
5 years ago, yeah, absolutely. Yes, they printed on the package,
But even that
even that
really mean that they're GMO
exactly.
free.
Well,
But
and depending on what country it comes from, they don't have to print it on the label.
exactly,
you know?
there's a whole, and that's
That's a whole nother topic for a
fun
whole nother thing.
conversation. But no, that's exactly it, though, and you can kind of every one of these conversations that stems from CRISPR becomes this huge tree of what if this or what if that.
Well,
and
you just said it. You're messing with the tree of life,
you are, yeah,
which is mind-boggling that we can even do that sort of thing.
right.
Uh,
We're at the point when we can create a species relatively easily.
Well, then does it become? Ooh, does it become X-Men? Does it become the outs? You know, like
That's going to happen. I mean,
Yeah.
it's going to.
that's
so let's
the
say,
that's the X-Men. You know,
here's kind of the thought I always have in the car. Always have. I do. It's a dorky thing.
Dog in the car. That's I get it all the time.
But let's say all of these, let's say the G7 countries, since they're in the news so much lately, let's say they all say, hey, we're not going to allow human modifications. We're just not going to allow it. We're going to wait 5 years, and then we'll
Yeah.
kind of readdress it then. It just takes one country, and they're going to do
Yeah.
it. And it doesn't even need to be a country. That's way past it. It takes one dude in his basement.
Because the
But-
cost of
but-
this is so low that it
Exactly.
We-
and it's getting
they
cheaper.
pose this in X-Men.
Mm-hmm.
They're the outsiders. You know, they're going to put them in a casting. You know, of a like, hey, you've got these genetically modified. You know,
Well, so this is a good point. What do you do then? Let's say some dude in his basement affects or kind of injects several stem cells with this. There's no time limit. It's going to be a gamete, and it's going to pass it on 100%, like we were talking about earlier, like the mosquitoes. They're going to pass hyperintelligence and hyperstrength off to all of their offspring, which are complex.
sure.
It's not just a one. I mean, this is 5-10 years
Oh, yeah.
down the road, which is not that long,
Right.
but we
or
get a
more,
little time
or
to
more,
work with.
hopefully. Yeah. But
Right. But let's say they do it. Then what do you do with those offspring.
Yeah. Do you segregate them from
And that's
society?
literally-
You create them in class. Do you
Suddenly we are the bad guys in X-Men.
Yeah. Do you kill them off because?
But they're going to pass it on to anyone.
Yeah, absolutely. That's the thing. Is like what's the ethical thing? We don't want to we don't have this is something that we do not want in society yet. We haven't created rules and regulations on
Right.
this. Do we segregate them and basically put them in like prison, basically?
It would, yeah.
Or
it would
do
be
we,
that.
Or do we kill them off? Is it ethically okay to kill them because we don't know
Right.
what to do with them? And there's so many ethical issues.
And we're going to have that conversation.
Absolutely. I think that oh, crap. There we go. I think that banning it creates a problem
Because it's not.
Yeah, no, you shouldn't
No
ban it
one's-
yet.
because if you ban it, then
I said that.
then it just goes to countries or
Yeah.
or somebody in a
an
basement.
all-out ban. I just don't know how you would ever.
No.
Again, you're not talking about
We've already come so far.
right. You're not talking about a complex. Again, if you spent a week, you could do what I've done in undergrad, which is create a beer that produces cinnamon flavor. It doesn't take that much. It's not
Right.
that difficult. If you can understand, you can understand CRISPR.
Yeah.
You have a beer that's cinnamon flavored?
So it turns out yeast that creates cinnamon flavor, which I thought was going to be this great invention that Oh, okay, not cinnamon, vanilla flavor, vanilla.
Either one.
Either one.
Uh,
I'm just saying, you have a
so
beer.
my thought was
why did that beer not
the
make
vanilla
it here?
bean infusion. Gosh, how much money do they have to spend on this vanilla flavoring?
Mhm.
If I could just create a strain of yeast that produces it, and this is before CRISPR, and we could do that pretty easily. The problem is though, the vanilla flavor kills off the yeast, so they excrete it and then kill themselves, and then obviously it's not very useful for it.
Exactly. Yeah, it doesn't impart
You can
the
glow in the
flavor as
dark
much.
beer without killing the beer.
Well, we did that with the zebrafish.
Glow in the dark beer? I don't know if
So-
I'd eat that.
You know those glow-
And then you use the restroom, you're like, oh, what is going on here?
Or you're like, this is the greatest thing ever. I'm so glad they put.
you know
Now I can see the toilet.
Yeah,
you-
this is
have-
so much easier.
have you heard of those the zebrafish that glow in the dark?
Yeah.
That was CRISPR, and they're $10. You can buy a glow in the dark fish for $10.
And that was, and that was early infancy of the.
Right.
yeah, that was way long ago. So it's already happening. It's a
Well, I mean, just down to the little dogs that we have nowadays. Those are CRISPR dogs for sure.
I don't know if they're
Crossbreeding and crossbreeding
Not
and
necessarily
crossbreeding.
CRISPR, but they're
Yeah.
certainly
GMO
genetically
dogs.
modified or altered dogs.
Since
Exactly.
you finished your beer from Fort Bryan, what would you like? Would you like a one of these again, or would you like the just the fairy nectar?
Let's do the fairy nectar.
Sorry, Brian, you're not getting any fairy nectar.
that's fine. So
On the bright side of things,
I
I'm very
still-
optimistic about all this stuff.
that's all right. I still have a whole cooler full of stuff over here.
Oh. Sorry.
Since you're tied more into the scientific community, I'm going to get into the how do we solve this.
Sure.
So we can't do a ban.
Nope.
Because all a ban is going to do.
Well, the question is, do we solve it? I mean, or do we let it
in
ride?
in a way. I mean, you need to. There are some safeguards that we need to put
Is it
into
a
place.
problem? It's not a problem yet, but it could turn into a problem.
I would
Very
guess-
easily.
So most people say it is not a problem now.
No,
I would guess
because it's in the testing phases right,
right.
now.
I would guess it's already a problem.
It also
and that
depends
we're
on.
close to, if not already past the time. when
It
we
also
could have
depends on
solved it,
where your
which is
line
negative.
is.
But-
where's your line?
Well,
I mean,
this is where we get to Gattaca.
Mhm.
If someone has a solution to, if someone comes out with this, and theoretically they're not there yet, they could be, probably
Mm-hmm.
not. But if I can say with certainty, your child, your embryo, will have a higher intelligence by 50%.
Would you
There's a
like to do that?
market for it? It doesn't matter if it's a small market. It just matters if there's a market. And money talks.
Oh,
and you're gonna, and that's all it takes. One of those, and you have Gattaca, which is you have a high system. You have a caste of rich people that can afford that,
Mhm.
and everybody else.
Because right now we have the technology to say you are predisposed to at 28%.
Sure.
To get
technology. We can
Yeah.
save them.
yeah. We already have that technology to give the percentages of what we are predisposed to.
Right.
Does that actually happen in your life? We don't know that yet.
Right. And
That's-
again, right, and that's all, and that is dealing with chances,
But that's right now.
and that's now,
Yeah.
and we're already there
Yeah.
with, well, maybe you'll get Alzheimer's, But you have people that are saying, I can cure Alzheimer's, and I can prove it to you that I can cure it.
Mhm.
Are you going to give us, $1 million for this cure for your embryo?
Well,
and there's going to be a market for that.
So, and that begs the question. The next step to this is CRISPR, once it becomes available to the public, is it only going to be the wealthy? The answer to
Well,
me
yes.
would be absolutely
yes, because it is what it is. I mean, TVs. You want the awesome TV? Well,
Yep.
I'm sorry, that's going to be $47,000.
It's like a menu.
Yeah, you can't. Yeah, it's a menu.
Mhm.
Now, over time, when it progresses, it's going to get cheaper and cheaper. And something that you could have gotten 5 years ago for $47,000, now you can only get it for $5,000. And
But-
people are like, "Oh yeah, I can afford $5,000."
But your analogy is talking about TVs. We're talking life. I mean, that.
I'm just saying, with
Yes, it will.
with more people buying it, the cheaper it will get over time.
And what I'm getting at is there are more ethics when it comes to life, because
So-
if the wealthy are only available to afford this, ethically,
Oh yeah, you got to pay all those doctors.
We're
'cause
also
of all the
assuming
research they've done.
these the wealthy want it for everyone else. The other catch is
Mhm.
if you're very wealthy, or if you're a very wealthy family,
Mhm.
and you have the ability to buy this out and control it on your own, suddenly it's not just necessarily the wealthy who have this access. It's whoever you say has this access
Mhm.
has this access.
exactly, and,
And-
then it creates another class of person, not just a wealthy person, but
Well, look at
an enhanced person.
look at politics. It's rarely is it this kind of up and comer story of Joe Schmo who
Oh, absolutely.
was raised on a farm and became a politician. It's generally connected people, and that's
that
what
are
you're going to get.
generational politicians.
Exactly, and there's a reason for that. It's that connection. It doesn't necessarily.
He's
It
got
usually
the, he's got
coincides
the
with wealth,
politician
but
gene.
It's yeah.
I don't know where they're going.
But
with
at the
that,
same time, do we want to explore Mars? Do we want to advance our species?
Oh
We
yeah,
have this
I mean,
capacity to.
you could land on Mars and not have to have a spacesuit because you're like
Right.
the gene for going to Mars. There you go.
Right. Suddenly we
done.
can survive in space a lot longer than we used to.
So, and this is going to be a little bit of a tangent on what we're talking about, but I just want to spend a couple minutes because
Tangents are great.
So I just heard something recently on this, and it was on the Joe Rogan podcast, and he was talking to Neil deGrasse Tyson, and he said, why have we not been back to the moon? And so Neil deGrasse Tyson's response to that was, the only reason we went to the moon was because we had motivation to do so. That motivation was either we beat Russia to the moon or we die.
Yep.
That was literally the motivation.
Or we
And
die.
yes, because we were at war with Russia, and
And if they beat us, then it was pretty much they win.
Even
Yeah.
an ego hit at that point.
Well,
Was a
yes.
Huge.
So yeah,
Okay, I see your point
more
there.
prevalent was the ego, but yeah, it was more of a we are at war with this country.
Right.
If we don't beat them to the moon, then they pretty much win, and then we die.
Right.
What reason do we have to go to Mars other than
Thing I said.
if we stay here on the Earth, then we die? So he said the reason we haven't gone back to the moon, there's no motivation other than there is a little bit of money tied to that. What minerals could we get from the moon?
Sure.
So
I'd go
which are
I'd-
minimal compared to asteroids or things like that.
exactly. And
Speaking of
that's-
asteroids, look at the moon. I don't know if I'd want to live there. Just it's,
well, you don't have to live there to mine it. I mean, you can send
no,
robots
but
up
it's
there.
it's
Yeah.
it's spotted with nice.
Right, yeah. Dark City, is that your tie-in?
No,
Palm
sure.
Beach.
Yes.
Oh,
yeah,
Yeah.
absolutely, sure.
That was the intention.
anyway,
Sometime.
so yeah, the asteroid thing was, but that was my small tangent. But
No,
on
but that's
the-
true.
No, it's
there
good
has to be a
stuff.
motivating factor.
which kind of ties into the motivating factor of how far we go and how soon we
Sure.
get there.
And it was a simpler time back then, right? When it was just government versus government. And nowadays it's just with globalization, which is not inherently a bad thing, but with the idea that we're kind of all on this theoretical level playing field,
Mm-hmm.
it's less about these governments going at each other, and it's more about these groups of people who have
But
wealth that
but
want
if
to
you're
fund
first,
things.
if you're not first, you're last.
Exactly.
Yeah.
Well, and especially with CRISPR, if you're not
Mhm.
the most intelligent, then you're what does it matter?
So back to, and we kind of got on tangent. I don't know what tangent we're on. So back to what I was saying with the scientific community. You're tied more into that. So what I want to know is, From a solving standpoint, I know that in the scientific community, there's a lot more safeguards on new technologies that are coming out. And yes, scientists are all giddy about
Right.
new stuff
Sure.
that's coming out, but
Certainly.
they're also like, oh, hey, we should probably hold off on this and do the self-imposed moratoriums and all that. So, from a solving standpoint, beyond an ethics committee, what do we need to do?
Well, that's the big question. I
$100,000
suppose.
or
Yeah, right?
$1 billion question.
So, and this is something I continually ask myself. So we talk about the wealthy having access to this over, just Joe Schmo
Mhm.
in Nebraska. And we mentioned it again earlier with mosquitoes, with this sex cell versus, somatic cells. Or is it going to be a temporary fix or a long-term solution? And the risk of, let's say I think I have a cure for stupidity. Let's say I think I can fix stupidity.
That would
which,
be
again,
awesome.
most people
Please, can you cure that?
theoretically would want
That's
to
awesome.
Resolve. But let's say I have a fix for it. We don't necessarily know that there's not going to be a side effect, like all of a sudden I die when I'm 20.
Mhm.
Something.
Is it the common sense?
Yeah, there's just
Can we
the
insert
world
the
is
common
too
sense?
much when you're that intelligent. So we want it. We kind of want to tamper that expectation by putting limits on how long this gene is going to last. So maybe
Right.
it ends in one generation.
More in the mic there.
But at the same
Okay.
time, to kind of counteract this idea of only the wealthy get the benefits, do you
Which is gonna happen.
which is
real-
going to. I mean, you're naive to think that it's not going to happen like that.
Mhm.
Do you force then the opposite, which is it has to be
available
passed
to
down?
everybody or nobody.
Well, even available to everybody, which won't you just can't legislate that.
No.
But you could easily legislate that if you put this in your kid, they're going to put it in their kid,
Mhm.
and that's going to pass down. And then all of a sudden, if they're intelligent, they're going to spread that intelligence gene. So theoretically, at least, the future of humans would either be intelligent or would die at the age of 20 for.
Right. And is this one of those? And I'm gonna go out way beyond our generation.
Oh yeah,
So is this necessary for the human race to explore this so we can edit ourselves to live longer to do space exploration to further the human race? Because right now, I mean, if you think about the sheer time frame involved in space exploration,
it's not doable yet.
no, I mean, we don't have the cryogenic freezing process or stasis available. I mean, yes, that's in sci-fi movies, but that's not a reality right now. I mean, we can put ourselves in stasis for a certain amount of time, but not 100,000 years to traverse one
Right.
galaxy. One.
It's insane.
Just
Yeah.
one galaxy.
We'd be lucky to get that far.
Yeah.
Assuming
So-
you don't hit an asteroid on the way.
Exactly. But if we have a generational ship where people are living thousands of years per person,
Right.
then it's
Suddenly-
then you're talking feasibility.
And I don't know. I guess that's where I become this optimist that
I
doesn't
mean,
care
it kind of
about any
sounds like
the side
Austin
effects.
Powers to me.
A little bit, though,
Well,
right?
I was-
You get this warp shit.
I was actually thinking more Star Trek.
Oh yeah.
I mean, you have a generational ship.
Yeah.
It's
which
Event-
they didn't really.
It's Event Horizon.
They,
Yeah.
Well,
Whoa.
that had to,
Just-
didn't that have to do with the being on the edge of a black hole?
Black
and-
hole.
Yeah, but they were all in space time. They were in cocoons,
Mhm.
and
Well, most space shows have or space movies have that where they're in some kind of stasis and all that. And is that even necessary?
cryostasis.
Yeah. With CRISPR, is it even necessary?
yeah.
Yeah.
I don't know. So as
I'm Yoda. I'm 1,000 years old.
right, and that'll be totally normal.
Yeah.
in
And
20
again,
years.
then that gets into religion aspect
If
also.
you've only seen what I have seen.
Yeah.
No, I don't want to see what you've seen.
We're good.
Yeah.
We're good.
Yeah, there's a lot of tie-ins in this that I want to do, like a part 2 that focuses more on the faith and religion aspect.
And I think that's a good conversation to have,
hmm
kind of, because it's going to be a conversation that we have to have.
hmm
And whatever side of this kind of aisle you land on, again, there's going to be someone in his basement who's going to do it regardless. Like,
Mhm.
no matter how much you try to mitigate that, whether you are on one side or the other, there's someone who's going to do it because they want to do it.
Oh, absolutely, and that's. I mean, we already talked about banning it, and banning it's not going to help because it you don't even. I mean, you could still be connected to the UN and say, you know what, I'm going to still do this in my basement. It takes me. It's $75 and whatever my setup cost for the lab.
Right.
It doesn't have to be a rogue.
$75. That's just, it's
Well, that was
astonishing.
That was the price 2 years ago. So it could be cheaper.
Going down.
Yeah.
It's like buying Tinker Toys,
Exactly. Yeah.
I can do that.
I'll get a Lego, I guess.
Yeah. So
Cheaper than Legos. That's
right.
where we're at right
Oh,
now. It is
man.
cheaper than a Lego
that's a
set
valid point.
to
It is.
change the DNA of a species
That is-
forever.
Wow, Lego, take notice.
Yeah. you gotta step the game up.
Yeah.
That plastic's expensive.
That's true.
All right.
No,
So that's pretty much all I had on this one.
Yeah.
There are so many avenues that we could go down, but
Solving the world's
Yeah.
problems, though. I mean, where we're at is, I to solve this, I think would be to on.
it's so
Go on
cheap.
and forth.
It's so cheap and easy right now. It's not going to be shut down.
Right.
Right.
Yeah.
Well, the benefit is
It's the
It's-
race to the moon.
Right,
We
it
are
is.
trying to be the next
Mhm.
people that go, ha ha, we've got it. We figured
Right.
it out.
and that's that's the future you want for the next generation
Yeah.
of anybody is a better life with
Absolutely.
less disease. And
Yeah. Like I said, unethical to not use it
Right.
in the future. Yeah.
Well, and how cool would it be? Like Wolverine, you get a cut, and you're like, oh man, I got to get a Band-Aid. But CRISPR is like, and you're like, oh no, I don't need a Band-Aid. I'm fine.
Oh, there was that new there's a new movie with Sam what's his name?
Goody.
He was in Avatar. And now there's a new movie on Netflix. And basically we're trying to genetically engineer human beings to go to Mars because
Sure.
Titan, the moon on Saturn. Is that right? Or Jupiter? Saturn.
Titan.
I want to say Saturn.
I'm just thinking of the movie Titan AE, and that
Yeah.
was a pretty good.
and I think
pretty
that-
good that was a good film.
And actually, I think that's the name of the movie, Titans. It's on Netflix, and they do a quick job. Like it's a very forced evolution, and it's extremely quick, which
Sure.
is not possible. But the atmosphere is not conducive to humans, so they're trying to evolve humans to
adapt
actually.
to that.
So you don't need a spacesuit on
Sure.
Titan. You can just
Go out.
go
Right.
out.
Yeah, just-
Which would be huge for terraforming.
Yeah, and that's kind of the future of CRISPR is evolving humanity into what we need.
Sure.
And the safeguards, though, it's just and it's on so many different levels.
Yes.
Is it per person? Is it generational?
Oh, and you
Is
guys
it-
are talking about going back to the moon. Well, you just create a person that's a lot heavier. We just send all the
Suddenly.
fat people there, and
Yeah.
then they don't bounce around.
mass doesn't
No,
work
that's
in
horrible.
zero.
I was horrible. on
Yeah,
that.
zero gravity. I mean, then the weight of the person doesn't matter in zero gravity.
But you could theoretically make someone less dependent on oxygen,
Mm-hmm.
which in turn makes it cheaper to space travel. You don't have to send as much
All right,
oxygen
science
up there.
teacher.
So,
Yeah,
the
exactly.
optimist, I
Yeah.
guess.
Right?
Yeah, or if you go to a world that is completely encapsulated in a liquid.
I
Pull-
think
pull
that
the-
all of
pull-
the
Oh, yeah.
positive.
We don't need
gills if we need gills.
Yeah, we don't need a nose. We need to pull oxygen from
Sure.
the liquid.
There is so many non-negatives and so many positives to this CRISPR thing, so solving it is, we're not solving it, we're
just going
aiding
to,
it, we're
Yeah.
making it better.
Yeah.
And thankfully, we have enough intelligent people that can, if something does happen that's negative, that theoretically they can CRISPR the solution to it.
I'm going to go with theoretically we have intelligent people.
Well,
there.
if we then there's that space race towards intelligent people where
Yeah.
everyone has to make the smarter human.
yeah.
So
Sure.
one important thing to note, kind of going forward too, is there's a lot of industries that are going to suffer from CRISPR.
Mhm.
Like you said, I mean,
nanotechnology.
Oh, but,
will suffer.
nanotechnology. would be one,
Medical companies will
but
suffer.
Band-Aid. If you can create
Antibiotics.
if you can create humans that heal themselves better, Band-Aid's going to go out of
Oh, yeah.
business. Antibiotics are going to go out of business.
Oh, I didn't think of
So
that.
all of a sudden, when you
great.
watch the news and you hear these news stories about CRISPR, there are going to, and rightfully so, be negative news stories. But you need to be cautious about
Why are they
is
negative?
backing
Yeah.
these stories,
Exactly.
because whoever's a giant producer of an antibiotic is obviously going to be against.
Which is a huge. You want to go up against a drug company?
Yeah, oh, yeah.
Really?
Well, we've had a cure for cavities for 20 years now.
Yeah.
We've had a cure for a lot of things for
Yes.
a lot of times.
But if there's no money in that, if it's
Yeah.
a cure,
Why do you want to
Why
cure
would
it
you
when
wanna
we
cure
could
it?
have somebody dependent on
Exactly.
medicine
And
for the
it's
rest of
gonna
their
be-
life?
It's why I became a teacher. The dependence on being able to, like, kind of digest a news article and really understand where it's coming from and if it's genuine or if it's got a kind of an alternate purpose behind it.
Right.
Is going to be huge for us.
Nice.
Yeah.
I think that's
awesome.
I think that's a
ending.
good ending right there. So we've been drinking on Kros-Strain, and we had the Pseudo Sue.
We also had the homebrew.
The homebrew from Andrew. Thank you, Andrew. It was
Yep.
awesome. We've been talking on the CRISPR Cas9 system.
So many possibilities.
Oh man,
Can't wait.
yeah. We kind of had the Cliff Notes version today. We went like we're coming up on 2 hours, but really, do a search. It's mind blowing what you will find.
Well, hours and hours and hours of research.
Yeah, exactly. It's coming down the pipeline, and I think that
The superhumans are coming.
yeah,
We're almost
Yeah.
there.
Yeah,
X-Men.
The GMO
So-
humans are
Yes.
coming.
yeah, we are so close. But yeah, this has been an episode of Drink Talk. My name is Brian.
I'm Britt.
Ted Nick.
Nick, all right. Thank you for joining us.
Yeah, absolutely. Thanks for coming on board.
Yeah,
Oh, yeah.
appreciate being the science guy.
Always,
So-
always.
All right. Have a good night.